Okay, good morning. It is a real pleasure for me to welcome all of you to this one-day symposium on lung cancer. As you know, today is the International Day of Cancer Research, so I think it's most appropriate to have this symposium organized in a day such as today. First of all, I would like to thank Fundación Areces for their support, financial and organizational. As you can see, we are in this wonderful auditorium, and they have really made the organization of this symposium really to work absolutely flawless. Obviously, we also, I also have to thank María Ángeles Santamaría and Álvaro Ferro, both from the group of Luis Paz-Ares at the 12 de octubre, October 12th. For those of you who are not in Spain, some of us, old enough, remember that it was October 1st, but now, from many years on, it's October 12th, the name of the hospital. And obviously, I would like to thank, special thanks to Professor Julian Downward, who has been kind enough to spend a little bit of his precious time in coming to Madrid and to tell us the the kind of fantastic work that he's been doing over the years. I also have to thank all the members of the project iLUNG, that actually was the basis for this for organizing this symposium, and in particular Marco Hermalumbres, who's done a lot of fantastic work in preparing this project that is funded by the Autonomous Community of Madrid, and that puts together people with different expertise, as you will see today. They will be introduced by Dr. Luis Paz-Ares. People with very different specialties, from nanotechnology to animal models to obviously clinical research. So I think, I hope you will enjoy the program, and you will think that it was worth coming to attend this symposium. Then finally, I would like to thank Manuel Azcona for his help in organizing this meeting. We also have the presence of Dr. Emilio Boza, who is the head of the scientific council of the Fundación Ramón Areces. So I think this is all I have to say. Again, thanks to all the people who have made this one-day Congress possible, and I really hope that it will be worthwhile, your attendance, and that we will learn about the latest research from different aspects in something as critical and as important as lung cancer. So now allow me to introduce Dr. Luis Paz-Ares. He's the head of the 12 de octubre oncology department, and he will introduce the speakers. So Luis.
So thank you very much, Mariano. Thank you very much for all of you for coming here for this first symposium of the iLUNG. iLUNG is a consortium that is being funded by the regional government of Madrid, and that is a project that gets together a number of investigators of different fields, from more basic bacteriology to physics, clinical pathology, and clinicians, medical oncologists, trying to get some better understanding and some answers for lung cancer, particularly KRAS-driven lung cancer and some others. So I'd really like to thank as well the Fundación Ramón Areces for allowing us to have this event here, and also to have the event translating the doors of the foundation by being online as well. And of course, I'd like to thank Professor Dongworth for coming all the way from UK to here today as well. So you see here what is very much I think, if I'm going, okay. So I don't know if I'm able to really, okay, here you go. So here is more or less the agenda we have for today. And as you see, we're having Dr. Sugaseoitia, who is a medical oncologist at the hospital Telho in Madrid, and he's going to tell us about the role of spatial transcriptomics and clinical applications in lung cancer. Then a more pathology-based lecture on artificial intelligence for lung cancer, how to guide the treatment with targeted therapies and immunotherapy. Professor Dongworth has been invited to give one of the keynotes talk in this event, investigating the interplay between KRAS signaling, oncogenic signaling, and tumor evasion in cancer. And Marcos Malumbres, also one of the other components of the iLUNG consortium, opportunities for cell cycle targeted therapies in lung cancer. Dr. Monica Mustiano on targeting KRAS-driven cancer. Dr. Javier Tamayo exploring cancer cells through the lens of physics, the role of optomechanical technologies. And finally, Dr. Usero on iron import and immune suppression in squamous lung cancer. It is important that all those talks are given by the relevant PIs of those consortium. And after the lunch, we will have the opportunity to have some 6 talks by more that we have selected from the junior people here, all as well, more or less within the people in the consortium or students of an international PhD student program we have between the University Complutense of Madrid, the University of Torino, and the University of Rotterdam. I have to tell here and thank as well all those universities for helping us on this program. And I have to excuse Dr. Trivio Brusolino that was in the program also as a keynote speaker. And due to a sudden personal issue, he's not been able to make it finally, but I'm telling you, it's really, it was really a big effort from his part to try to come, but it was not possible again. So I think without further delay, maybe we could start the event. I'm just going to talk, I'm going to ask Dr. Sugaseoitia to come to the podium to start this morning. So thank you very much.
Okay, thank you, Luis, for the introduction, and thank you to the organizers for inviting me to share this talk with you. As Luis was saying in the introduction, I'm going to try to explain how we could potentially use spatial transcriptomics to answer clinically relevant questions related to lung cancer. I'm a medical oncologist working in October 12 Hospital, and I'm also doing some research in the lab dedicated to identification of biomarkers and mechanisms of resistance to targeted therapies and immunotherapy in lung cancer. So this is my disclosures. And as all of you in the audience know, so there has been a major breakthrough, or perhaps I would say a revolution in spatial omic technologies. This is a review published back in 2021, a couple of years ago, where we could literally assess perhaps 100 of genes or maybe 1,000 of genes in a spatial context. And nowadays, currently, we are able to assess the entire coding genome at subcellular resolution in a spatial context. So really a major advance at the technological level that can certainly be used to answer fundamental biology related to how the spatial context can affect fundamental biological procedures. But the question still stands to whether these technologies could be applied or not to answer clinically relevant questions, or meaning whether these technologies will ever reach the clinic or not to improve what we have in terms of developing diagnostic tests or trying to answer questions that are relevant to patients. And this is what we are trying to address in the lab. And I'm going to show you how we can apply these systems to answer clinically relevant questions. And we use two key examples for that. One, how can we use spatial transcriptomics to decipher intratumoral heterogeneity, and why and how can this be important to assess treatment outcomes in small cell lung cancer. And then I will use another example of how can we use these technologies to develop predictive tests in order to forecast what is going to be the outcome of a given therapy to a particular subgroup of patients. And in this case, I will use an example of EGFR mutant non-small cell lung cancer. So in the first key example, as you know, small cell lung cancer is a very unique type of lung cancer. 10% of, or 5 to 10% of the lung cancers that we see in people, and is characterized by being very aggressive, and typically at the biological level, it doesn't have or it doesn't contain mitogenic drivers that we can use to guide therapeutic decisions in these people having small cell lung cancer. Back in 2019, as we don't have these mitogenic drivers to subclassify the disease, these authors were proposing that we could instead use transcriptomic analysis to subclassify this disease. And as you can see, these authors were proposing 4 subtypes of small cell lung cancer based on underlying transcriptomic profiles that could be potentially relatively easily accessible by interrogating the expression of 4 key master regulators, as you can see there, ACL1, NeuroD1, PO2F3, and JAP1. And this was important biologically, but also the authors were proposing here that these 4 subtypes could potentially contain, or at least that the expression of key genes could be subtype specific, and we could potentially use these subtypes to guide therapeutic decisions in the clinic. And this was taken very positively from the oncology community, because this was kind of the first time that we could potentially subclassify small cell lung cancer in groups that could be potentially clinically relevant, and this was actually very good news back then. And actually, a couple of years later, this was this article published in Cancer Cell that we could potentially use this transcriptomic subtyping to identify people that were responding better to immunotherapy. This was one of these transcriptional subgroups, was characterized by having a greater underlying inflammation intratumorally, and those patients apparently were doing better when treated with PD-1 axis blockade. So we wanted to analyze this further, and we collected tumor samples from patients treated with chemotherapy and immunotherapy with small cell lung cancer. These were divided into different cohorts, and we wanted to address how these transcriptomic subtypes were being represented intratumorally, and interrogate also how was this, or assess potential intratumoral heterogeneity in these transcriptomic subtypes in human small cell lung cancer. And for that, as I was saying, we collected retrospectively tumors from patients treated with chemotherapy plus immunotherapy. These are two examples of baseline tumors, meaning tumors before starting any kind of therapy. And as you can see, well, there to assess these transcriptomic profiles in space, we are using a system called Geomics Digital Spatial Profiling that basically you can use it to select regions of interest within the tumor, and you can assess in those particular regions gene expression. And for that, we were using an assay that contained a total of 1,800 genes. As I was saying before, you can now assess the entire transcriptome, but back then we were assessing 1,800 genes, and you have the profiles of these genes expressed in these different regions of interest, and these are two examples of tumors from patients. And as you can see here, we could, by using this technology, we could essentially reproduce these four transcriptomic subtypes quite nicely in terms of distribution. We found that we could actually detect these four subtypes, and the distribution was very much comparable to what was published back in 2019. But importantly, when we assess these transcriptomic subtypes in each of the regions of interest, we realized that approximately 50% of the patients have, let's say, homogeneous tumors in terms of these transcriptional subtypes. Each of the regions contain essentially the same subtype, but the other half of the patient actually, there is a mixture of different subtypes being co-expressed in the same tumor. And this happens even before the start of any therapy. So 50% of the patients actually don't have a unique subtype, but different subtypes can coexist in the same tumor. And probably this heterogeneity is even higher, because as you can see, there were a number of patients here where we could only assess two regions of interest, and the more the regions we were assessing, the more heterogeneous the tumors were. So in the end, this speaks about how complex these tumors are, and also speaks about the limitation of these classifications to impact clinical care because of this high heterogeneity. And not only that, but these targets that are potentially clinical targets do not show actually a complete subtype specificity. As you can see, for example, DLL3, which is a target that we are now using or potentially using as a target for T-cell redirection strategies, there is a predominant expression in this subtype A, ACL1, but as you can see, virtually all tumors express DLL3 at some level, which means that subtyping actually doesn't really matter that much to potentially select a therapy based on DLL3. And the same happens, for example, for B7H3, another target that is being very closely monitored as a target for antibody-drug conjugates. And as you can see, the expression actually is not any different between these four subtypes. So in the end, this heterogeneity, coupled with the fact that there is no complete subtype specificity, actually limits the application of this subtyping in the clinic. And we could assess this because we were interrogating these subtypes using spatial methods. So this is more or less an example of how these technologies can be used to decipher intratumoral heterogeneity and why this can be important for clinical decisions. So we now move on to the second example, which is a totally different thing, EGFR mutant disease. This is lung cancer, but of course it's a totally different disease, as I was saying. So as you know, EGFR mutant lung cancer represents about 10% of lung cancer that we see in the clinic, typically associated with non-smoker, totally opposite disease as small cell lung cancer. We have very effective drugs to control this disease, osimertinib being the current standard of care in the advanced setting. So osimertinib is an effective drug to control advanced EGFR mutant lung cancer. But as you can see, this treatment is actually not curative. And in fact, actually, most patients progress within 2 years of therapy. So it's a good drug, but we need to do better. And importantly, despite the fact that the vast majority of patients actually respond initially to the drug, there are even some patients that do not respond at all, or responses are short in time. And because of that, there has been some clinical investigations on trying to escalate osimertinib monotherapy and see whether we can, either using chemotherapy in combination with osimertinib or using another combinatorial strategy based on lazertinib, which is a similar drug to osimertinib, plus amivantamab, which is a drug that blocks EGFR and MET, is a bispecific antibody. You can see here in these curves that escalating therapy can help patients. Actually, you can prolong the time that patients remain with their disease under control. But of course, this comes to a cost of a greater toxicity. Combinations, it's not the same to give osimertinib as monotherapy or giving osimertinib plus chemotherapy. You are going to give chemo-related toxicity. And in the case of amivantamab, you will start seeing MET-related toxicities. And the question here is, do all patients require combinations? In which can we safely avoid them? And also, which one is best for the optimal frontline combination approach? So with this in mind, we were actually hypothesizing that maybe developing a test that can predict or identify beforehand which patients are going to progress within the first year or within 10 months, being these patients of high risk of rapid progression, could be potentially those where escalating therapy could be more reasonable. And for that, we again collected tumors from patients with advanced EGFR mutant lung cancer. There was a multi-institutional effort. We were able to collect approximately 100 of tumors. This is the baseline characteristics of the cohort, which basically resembles what we typically see in EGFR mutant lung cancer in the clinic. And to define early disease progression or early resistance, we took the 10-month cut point just to identify a population that we consider is at high risk. And as you can see, up to 30% of the patients progress within 10 years. So this is not anecdotal. This is actually happening in 30% of the patients receiving osimertinib as monotherapy. So to try to develop this test, we were really focusing, again, in spatial transcriptomics. The field of EGFR mutant lung cancer has been very much focused in genomics, but genomics don't really tell the whole story, and actually there is a plateau in what genomics can actually tell us in terms of prediction of treatment outcomes. So we were really trying to move to transcriptomic analysis, and as we have this system now that can actually assess transcriptomic profiles in space, we were using it again in these tumors. And in this case, we were also interested in trying to assess whether stromal components, in addition to what the tumor cells themselves can tell us, whether the stromal components were actually also predicting or influencing treatment outcomes, let's say. So for that, instead of only selecting the region of interest, each of these regions of interest was divided in 2 different compartments. In green, the tumor cells, and in red, the tumor-associated stroma. And by doing these compartments, you can again assess the same panel as before, 1,800 genes, but separately in the tumor compartment and the stromal compartment, in order to see what is the stroma adding or not to this prediction of treatment outcomes. These are some numbers of the number of genes that we are assessing, and also the number of tumor segment and stromal segment that we are assessing. As you can see, this is a high-dimensional data set because, we have 100 of patients, but in total, as we are assessing tumor and stroma, we have over 1,000 samples in the end, and out of those samples, we are assessing 1,800 genes in each of them, so really quite large data set. And this is a summary of some of the results that we are getting. I will walk you through the table a little bit. So in order to develop these predictive models, we were using either logistic regression or more advanced adaptive elastic net regression models, and we are testing their performance in this single cohort using repeated internal cross-validation. So first, we wanted to interrogate whether clinical variables are actually capable to predict treatment outcomes, and as you can see, they are. So the performance status, which is how the patient is feeling symptomatically, and the presence of baseline brain metastases are actually two variables, clinical variables, very easily accessible in the clinic that actually predict treatment outcomes. This is not surprising for clinicians. We know that these patients do worse, and we do see that their predicted performance is about an AUC of 0.70, which is good, but definitely something that we could potentially improve. We also tested single genes, and we selected MET because MET is a known driver of resistance to osimertinib. And as you can see, MET by its own, MET expression levels are capable to predict treatment outcomes, but actually very modestly, worse even than the clinical variables alone. So then we took we generated these models using only gene expression, and we tested separately genes expressed in the tumor compartment, genes expressed in the stroma, and generating a final model containing tumor and stroma information. And as you can see here, when we use only the genes expressed in the tumor, we are able to achieve a predictive performance on 0.74, slightly better than clinical variables, but not too much better, to be honest. And something interesting is that although we are able to generate models using stromal information alone, meaning that stromal components perhaps are also influencing treatment outcomes, but their performance in terms of prediction of clinical or treatment outcomes are actually worse than when you focus on the tumor compartment alone. And if you combine tumor and stroma, it's not any better than the tumor compartment, meaning that perhaps the main focus here is the tumor cell itself. And finally, we were able to create a final model containing genes in the tumor compartment with these two clinical variables. And by including this model, we are able to increase the predictive performance up to 0.84, which is reasonable or at least promising enough to try to validate this in an independent cohort, which is currently exactly what we are now doing. This is the model. It contains 19 genes, 2 clinical variables. So I can tell you that there are genes related to cancer cells themselves, but there are a number of genes, a number of genes related also to anti-tumor immunity and especially innate and adaptive immunity as well in the model. Perhaps half of the genes are actually immune-related genes, and perhaps it speaks about the importance of immunity in predicting or at least in achieving durable responses, even in tumors where we are not giving immunotherapy, we are giving targeted therapy, but the underlying immune response might be relevant as well to achieve durable responses even in these oncogene-driven tumors. And finally, we wanted to really ask a question that we think is relevant, and is how does this spatial context actually does any better or not to the current bulk RNA methods that we have in the clinic that are more easily usable in the clinical context. Does this spatial context is any better to bulk RNA analysis? So, well, for that, we don't have, unfortunately, bulk RNA information of this cohort, but we just try to simulate a bulk mRNA analysis, what we call this pseudo-bulk RNA model. I'm not sure if the name is correct or not, but we just simulated this bulk RNA expression data by summing up tumor and stroma gene expression and trying to have a single bulk RNA value per gene in each of the tumors. And we realized that actually this model performs worse than adding this spatial context. So it might be that considering the expression specifically in the cytokeratin positive tumor cell compartment actually does matter, but we are, as I said, trying to validate this further and see whether this is actually real or not. So three key messages to conclude. First, spatial transcriptomics can potentially resolve clinically relevant questions, as I just showed you. A spatial context could be relevant for biomarker development purposes, but still, to be honest, there is still unproven whether the spatial outperforms bulk analysis as a clinical test to forecast treatment outcomes. Thank you. Well, I'm acknowledging here people from the lab that did the work.
Right. Okay, thank you. So I think, I don't know if there is any question. Carlos, Marcos?
Hi, Jan. So I think DLL3 is a good gene to focus on, given the new therapies, etc., right? And the question whether there are few cells which are negative for DLL3 is still very relevant, right? So in your data, you are proposing that even the non-neuroendocrine tumors are DLL3 positive, for instance. But I saw in the graph, for instance, the P subtype, the distribution is like bimodal, right? So you have a cloud of positive and a cloud of negative. So I guess the question is, since you have spatial data, can you tell, maybe the resolution was not good enough in this test, but can you tell whether there are few cells which are negative might be driving resistance to the therapies?
Yeah, so, well, the system that we were using, as you said, doesn't have like this single cell resolution. This is more a region or compartment-based profiling that we are doing, so we are not really able to, let's say, quantitate how many individual cells are positive for DLL3 or not. But it's true. I mean, it's clear that the neuroendocrine subtypes do express more DLL3, but as a clinician, I also care about the others that are non-neuroendocrine and can express DLL3 as well and could potentially benefit from DLL3 targeted therapies, same as or maybe less than these other neuroendocrine subtypes, but there could potentially benefit there. So the question here is, again, we are speaking of continuous markers which are very difficult to translate them into a clinical test. It's not a genomic marker that it's binary, it's present or not. The question here is that DLL3 is expressed continuously, and there are tumors that are non-neuroendocrine that could potentially benefit from these therapies as well. And actually, we do see this in the clinic. I mean, there are some data emerging on the potential to use DLL3 to select T-cell engagers, for example, and apparently they are not robust enough, and potentially because other tumors that express low levels or lower levels could potentially respond as well.
I wanted to ask you, what do you think about single-cell sequencing? Do you think is that ready for translational research? What are your thoughts as someone has worked a lot in spatial transcriptomics?
Yeah, so I have, I would say, mixed thoughts on that. So, I mean, now you can literally do single-cell transcriptional analysis or single-cell sequencing in space, even at subcellular resolution. And I think those technologies are good, at least now, to answer some fundamental questions that you really need those technologies to answer those questions. But for thinking about translational research or research that is oriented to developing a clinical test, I'm not sure they are totally ready yet. I mean, perhaps the way I see them is more using a more non-subcellular resolution test or assay that you can use to screen and identify perhaps biomarkers, and then as a complement use single cell technology to see whether, interrogate that biomarker a bit further. But I don't think they are ready to do large discovery type screenings in terms of biomarker discovery. But of course, I do find them very useful for addressing questions that are related to developmental biology, spatial biology, and so on.
So, John, looking at the second example, you got a signature that is helping you to predict the outcome of patients pretty well, particularly so you're able to identify those patients at high risk, which are, in theory, the candidates for combination therapies. So the question is your signature predictive or more prognostic? That is always something that we never know. So I suppose, how are you going to validate that? The second question is, looking at those genes that you have in the signature, could you predict maybe cisplatin-based chemo is going to help, or more a MET or a comprehensive EGFR blockade?
Yeah, those are actually the questions that we are trying to address in these validation sets that we are assessing. So the first question is particularly important because this signature might very well be only prognostic, meaning that you just identify a population of patients that do worse regardless of whatever you do with them, which is not a good news for a clinician because in theory you could potentially escalate therapy in those cases, but maybe this escalation is not going to help them too much, or at least is not going to help clinicians decide whether to use a single agent therapy or escalation strategy. So to address that, we have actually two validation sets. One is another osimertinib treated cohort, and the other, we collected a second cohort of early stage tumors that have not been exposed to TKI therapy. So in that way, we will be able to assess prognostic significance. Of course, as this is not a randomized, this is not a randomized cohort, so we will not be able to do this interaction test and so on to try to fully answer that question, but at least having this osimertinib untreated population, we think that could potentially give us a clue about their prognostic significance. And this is the only tool right now that we have, but we will certainly use it to see whether this is true or not. So,
So by looking at the list of genes you're getting the signature, what is your smelling about they are going to do better on EGFR blockade or on chemo?
Yeah, well, something that I can say is that MET is part of the signature. I just showed you in the table, but I'm not sure if this actually means that they are going to do better with MET blockade. We are also collaborating with Geoff McIntyre, trying to assess some of the signatures that he or his group developed in terms of predicting platinum sensitivity or capacity or to acquire MET amplification in the future, and we are actually testing that. And well, initial data suggests that perhaps half and half would be potentially more suitable for platinum-based combinations, the other half for MET, but still based on a very small proportion of tumors that we were able to assess that.
Any other question?
Hi, John, thank you. You mentioned that some of these genes are related to immune response, innate immunity, etc. And your stroma compartment were not very useful to improve the model. But is there any data on how, I don't know, EGFR mutant tumors are not too infiltrated by immune cells, but did you have the chance to check if maybe the density of CD45 cells could be affecting this prediction?
Yeah, so the composition of, the immune cell composition apparently wasn't affecting that much. At least the quantification of total CD45, total T cells, and so on wasn't really affecting that much. We already looked into that. But there are other components related to innate immunity and cytokines and so on that we do see that they apparently make an influence on predicting outcomes. And this has been kind of suggested before in other contexts as well, KRAS mutant tumors. I mean, there is building evidence that you really need some kind of pre-existing immune activation to achieve durable responses. And apparently this is what we are seeing. It's not that much of how many or what is the composition, but perhaps also the functional orientation of the ongoing inflammation and so on might be relevant.
There is no other questions. I think we can go to the next speaker. Next speaker is Dr. Fernando López-Ríos. He's a molecular pathologist, particularly expert in lung cancer, and he's going to tell us about the role of artificial intelligence of helping to guide immunotherapy and targeted therapy.
Good morning, everyone. Thank you very much for the organization and the invitation to be here today. So I have to admit, I'm not very fond of artificial intelligence. So that would be my main disclaimer. So I try to keep my friends close and my enemies closer. So that's why in the next few minutes, in fact, I will try to convince you or share with you 5 opportunities, real opportunities that we have for the use of artificial intelligence, trying to identify which patients are going to benefit from both targeted therapies, but also immune checkpoint inhibitors. So I'm going to try to use artificial intelligence for the estimation of tumor content, also for the identification of actionable targets, scoring PD-L1, I think is also another great choice. And finally, 2, I think, very new things, which is the role of tumor infiltrating lymphocytes as predictive or prognostic factor in patients with both advanced and early stage. And finally, the use of artificial intelligence for assessing the pathological response after neoadjuvant therapies. So the reason why I said I was not very fond of artificial intelligence is because I think I pretty much agree with some of the conclusions that were released in this editorial not long ago. And I think we are trying to apply artificial intelligence without trying to understand what's a real clinical question or why is artificial intelligence helping us trying to improve the life of our patients with lung cancer. So I think we need really more knowledge to oppose that initial tendency. And also we need, I think, a more structured debate to be able to use it, I think, in a more sensible way. So in order to do that, I'm really convinced that we need a more comprehensive perspective of the whole journey of a patient with lung cancer, regardless of histology, regardless of stage. So this figure depicts the journey of a lung cancer patient from a biomarker perspective. And it's super complex because you have to first diagnose the patient, try to classify the tumor, then you have to maybe run a few biomarkers using
simple immunohistochemistry stains. Then you have to organize comprehensive genomic profiling, ideally in all patients, again, regardless of stage, and in a reflex manner, and using next-generation sequencing to be truly comprehensive. And the good news is that many of our patients now are diagnosed in earlier stages, so we have also the options of adjuvant or neoadjuvant or perioperative strategies. So you might end up with a situation where you have sometimes to repeat this effort along the course, the clinical course of a given patient. And also, as John was telling us, unfortunately, some of our patients, or many of our patients, relapse. So we have to understand also those resistance mechanisms, which are now new opportunities for treatment. And this is only possible if we have a teamwork approach with very effective communication with everyone involved. So I have chosen 5 specific opportunities for artificial intelligence to make this whole workflow leaner, easier for clinicians, for everyone involved, for patients as well. So let me show you. So the first one would be the estimation of tumor content. So I'm going to try to show you a rationale, because also I don't have a lot of time to go into all the details, and finally an outcome. That again, we are using these tools in the clinic, so I think they are realistic. So this is very obvious, but I think not everyone is convinced, and the idea is that most of the technologies that we use really depend on the quantity and quality of the DNA and RNA input. So this is a very old paper, but I still enjoy reading it because it involved many, many thousands of patients. And as you can see there in blue, the performance, in this case, it was a DNA-based large NGS panel. The performance of that panel was clearly linked to the amount of DNA. And as you can see, if you have a lot of DNA, more than 250 nanograms, almost 100% of your patients are going to receive a valid result, versus if you only had between 50 and 100 nanograms, only 2/3 of your patients will have a valid NGS result. So this is not easy to do in the clinic, and to give you also a hint of where we are right now, the panel that we are using now in the clinic needs only 10 nanograms of DNA and 10 nanograms of RNA. So huge improvement in the last 6 or 7 years. But still, we do need a fair amount of DNA or RNA. So this is very subjective, how we used to do this in the past, and we still use, is to just look at samples, H&E samples under the microscope, and try to see if the percentage of tumor cells is above the limit of detection. But what you have here on the screen is one of these AI algorithms that can do this in a matter of seconds. It's still not completely validated, but as you can see there, the machine is perfectly able to separate the tumor compartment from the stroma. Also, it can identify areas of necrosis, etc., etc. And at the end, it tells you what the percentage of tumor cells are in your given sample. So very useful. I think it's a great help, particularly in those samples that are very close to the limit of detection, to that threshold. The second opportunity would be to try to identify actionable targets just by looking at samples that have been stained with hematoxylin eosin. So why do we need this? I think everyone is aware that what we have to do now in real life is try to annotate these Venn diagrams. What I'm trying to say is that the number of actionable alterations, particularly in patients with non-small cell lung cancer, is huge. For me, it's probably the most complicated solid tumor. And not only that, but we have to look at simple variants, complex variants. We have to look now also for a high-level amplification, and of course, actionable fusions. So we have the whole spectrum of oncogenic transformation in just one single tumor type. And finally, this is complex because some of these actionable alterations are not very frequent. And I think that's a very optimistic statement. They're super rare. Some of them are present in, let's say, 0.1% of our patients. So you have to screen 1,000 patients to find just one single patient, for example. And it's not uncommon that some of the very successful therapies are actually used in only 1%, 2%, or 3% of our patients. So again, very complex biology, and I think also a huge clinical and testing challenge. So why not use some of the morphological changes that tumors have to try to predict what's the actionable target that has actually caused that morphological change. So this is not new, and in fact, I have to tell you that sometimes, just by looking at H&E, we are right. For example, we now know that tumors that have a lipidic pattern of growth are usually those that contain EGFR mutations. That has been known for almost 20 years. We also now know that tumors that have papillary patterns or micropapillary patterns are the ones that contain, for example, RET fusions, EGFR mutations, BRAF mutations. So whenever we see a papillary pattern or a micropapillary pattern, we have to be very, very persistent because in many cases, our patient is going to contain an actionable finding. And finally, mucinous features are linked to, for example, NRG1 fusions or KRAS mutations. And what's most intriguing is that whenever we see signaling cells in lung tumors, that's a very, very good predictive marker for the presence of ALK fusions, ROS1 fusions, or even RET fusions. So if a human eye can easily identify these morphological changes, why not use artificial intelligence? So this is still work in progress, but there are several groups using different technologies that I think have come up with very reasonable sensitivity and specificity. We are currently working in the field of RET fusions. Unfortunately, I cannot show you results today, but I think we have very promising results. So I'm really confident that some of these AI algorithms are going to be used in the very, very near future to screen our patients for the presence of actionable findings, particularly those that are not very frequent, and I think it would be a wonderful way of increasing the likelihood of finding treatment, and also a good way of probably coming up with more cost-effective approaches for this comprehensive genomic profiling paradigm. The third possibility is why not use artificial intelligence in the field of immune checkpoint inhibitors for scoring PD-L1. As you know, PD-L1, it's probably now the only compulsory biomarker that we need to test in all of our patients, trying to sometimes decide if they are going to respond or not to immune checkpoint inhibitors. And I think you're also aware that this is a very time-consuming and, how shall I put it, very delicate task, because sometimes the cutoffs are very low. The cutoff for some indications is only 1%, so that's very difficult for the human eye to establish. So the rationale for using artificial intelligence algorithms here would be to try to bridge the experience gap and also reduce the turnaround time. Because again, I can tell you that this takes sometimes hours to come up with the right result. So as you can see here, on those two linear graphs, using artificial intelligence, there's really no difference between trained pathologists and untrained pathologists. And not only that, but also the turnaround time for scoring decreases tremendously with the use of artificial intelligence. So we are trying to generate now some data, because although this is very obvious, not everyone is convinced. In the field of artificial intelligence, there are also doomers, people that think this is not worth the effort. But as you can see here in yellow, when we use one of these algorithms, we are faster when compared to manual scoring. And manual scoring is those blue or purple dots. Observer number 1 was not very experienced, and observer number 2 was very experienced, but even if you're very experienced, the AI tool is much faster. So we think that faster is always better for the clinical team, and of course, we tend to think that this is better for patients. And so we are faster scoring PD-L1 with AI algorithms, but you not only want to be quick, you also want to be right. And also we were intrigued about the use of PD-L1 AI algorithms to see if we could identify more patients. So this is what this group did. They took samples from an important trial, and they used an AI algorithm, and as you can see there, they found more patients, even at very low cutoffs. 22% more patients at the 1% cutoff, and 4% more patients at the 50% cutoff. So this is huge. So in theory, AI algorithms could have clinical utility, which is what we are all trying to demonstrate. So the outcome here is still work in progress, but I wanted to show you a case. This is a real case from our practice. This was a patient with an early stage non-small cell lung cancer, as you can see there. And I was the one who scored this slide, and I thought the patient was completely negative for PD-L1. But to my surprise, the algorithm told me that the slide contained almost 5% of tumor cells that were PD-L1 positive. So I went back to the slide, because no one likes to be wrong, and I looked at the slide, and there were some macrophages there on the far left. So that's okay, because that's the internal positive control that I use to make sure that the stain has worked. But then there were also some cells in the center that were beautifully stained with PD-L1. And when I looked closer, I saw that membranous staining, so that was a mucinous adenocarcinoma with indeed some PD-L1 positivity. So the algorithm was right. So what I'm trying to say is that these algorithms are faster, and I think can help us identify more patients if they are correctly validated and used. The fourth opportunity that we can have in the space is trying to use tumor-infiltrating lymphocytes as predictive factors for immune checkpoint inhibition, or as a signature, as an immunoscore that could complement the current staging classification, the TNM. In this case, I'm going to focus on the prognostic value of tumor-infiltrating lymphocytes. The rationale here is that the density of tumor-infiltrating lymphocytes in patients with early stage non-small cell lung cancer has prognostic value. We know this for now more than 10 years, but unfortunately it is still not incorporated in the staging classification of patients nowadays. But as you can see there, in the line that is pictured in light blue, if you have a lot of lymphocytes, you do better. And what I mean by that is that after 6 years, after surgery, more than or around 80% of patients have not recurred, versus if you have very few lymphocytes in your tumor, around 30% only 30% of patients are going to do well after 6 years. So I think the difference is huge. The other important thing to consider is that we need to take into consideration not only the density, the raw amount of lymphocytes, but also the distribution, the spatial distribution of those lymphocytes. So, because maybe it's not the same if those lymphocytes are around the tumor, in the stroma, or inside the tumor, but only in the stroma without actually permeating the epithelial nets, or if we have lots of lymphocytes inside the tumor. So again, as outcome, this is still work in progress, but we have, I think, interesting data coming up. I wanted to show you a clinical case so you try to understand the clinical need, and I think the potential use of some of these algorithms to be incorporated for this predictive and prognostic challenge. So our patient was male, 69 years old. He was a former smoker, and he underwent a lobectomy because he had a tumor in the right upper lobe, and he was diagnosed as a stage 2B adenocarcinoma. So this is not super great news, but I would say, you know, the prognosis could be relatively good. And to our surprise, the patient had a very early relapse within 3 months and developed bone metastasis. So when we look at the tumor, we were really surprised because the tumor was very low grade. It had a lipidic pattern of growth, so that looks like one of those EGFR mutant adenocarcinomas that John was showing us before. So really surprised of this not very good outcome. And when we looked at the amount of tumor-infiltrating lymphocytes, we are trying to use the heat maps. So we look at these 3 different compartments. So in the first compartment, the peritumoral stromal compartment, I would say that this density of CD8 positive cells was rather high. It was even higher if we looked at the stromal compartment within the tumor, in the center of the tumor. But unsurprisingly, the amount of lymphocytes that were actually permeating, that were actually infiltrating the tumor nest was very low. You see only 23 CD8 positive cells per square millimeter. So that's super low. So just guessing, but maybe one of the reasons why our patient had a very early relapse was because the number of tumor-infiltrated lymphocytes were not really inside the tumor nests. And finally, and I think this is probably one of the last applications of AI tools in real clinical practice, is the assessing of pathologic response after neoadjuvant therapies. So this is the rationale for this challenge. So we now know that after neoadjuvant therapies, maybe pathologic response can be used as a surrogate marker What I'm trying to use is that we don't have anything better, anything that gives us an answer that is fast. So it could be a predictive / prognostic marker. There are some recommendations out there in the clinic, as you can see there, depending on the amount of viable tumor, patients do better or do worse. The classical cutoff is 10%. So if you have less than 10% of tumor cells after a neoadjuvant therapy in the surgical specimen, that patient is in theory going to do very well versus patients that have more than 10% of tumor cells after these neoadjuvant strategies. And I particularly like that Kaplan-Meier curve because I think in a very elegant way shows that this is not a binary decision, that in fact this percentage can be quantitative and patients can do extremely well if you have between 0 and 5% of tumor, or can do extremely bad if patients have more than 80% of tumor cells after neoadjuvant therapies. The way we do this in the clinic is we take the whole tumor, we section the tumor, we include all those sections for histology assessment, and we use a calculator, which basically is an Excel file, where we have to record the percentage of tumor, the percentage of necrosis, the percentage of stroma. So believe me, this is very, very painful and very time-consuming. So we've tried to compare this manual evaluation with the use of some AI algorithms, and we are quite pleased because we had at least moderate agreement between manual assessment and machine-assisted evaluation. So that's why we try to think of this as a reasonable agreement. Still work in progress, but I think this can be tremendously helpful to try to come up with more standardized ways of assessing who is going to do better, who is going to do worse, or who is going to need adjuvant treatment after neoadjuvant strategies, for example. So, in summary, I think we have to welcome a new member of the team, which is the AI algorithm. It's all about teamwork, really. We have internal meetings in the lab where we try to integrate all the clinical, pathology, and biomarker data, trying to make sense of so much information. But we also have to discuss all these results with our clinical colleagues in more conventional, more formal gatherings, clinical tumor boards and molecular tumor boards. And I think we are going to see more of these AI algorithms discussed at any of these meetings. And with this in mind, I wanted to thank everyone in the team for all their efforts trying to make sure that most of our patients have as much information as possible. And I also wanted to share with you the image of the new building in our hospital that I think we are going to move in there very soon now. So hopefully it will also, I think, facilitate many of these challenges. So thank you very much.
Thanks, Fernando. I have a very simple question, because I haven't seen many talks in AI in pathology, but the few that I've seen, most of the times based the analysis on H&E. Is anybody trying something different, like showing to the system, to the artificial system, a different stain that might improve the result?
I think when someone starts saying, oh, this is a very simple question, you start to think it might not be a simple question. I think, Alvaro, this is a great question. I think we have started using H&E stains because it's what we have. I think also we have, I would say, in the last 20 years, forgotten on how to look carefully at the characteristics of tumors, at H&Es. Before, in the past, we didn't have all this technology. We didn't have immunohistochemistry. We didn't have genomics. So we used to classify tumors only based on the characteristics of the membrane, on very subtle things. And in order to do that, you need very thin sections, and many, many times not stained with H&E, stained with other things, with other silver-based stains and lots of other histochemistry options. So I think now that AI algorithms need to really generate data based on some of these very subtle things, sometimes invisible to the human eye, or only visible if someone tells you, look there, I think we're going to see, we are going to reclaim the use of histochemistry and the use of other stains. Honestly, I try to think that that would be a good way forward.
I got another question. So we have seen over the last few years that ADCs are coming into the clinic, and for most of the cases, ADCs are targeting antigens in the surface of the cell, and the expression of those antigens in the surface of the cell, of the tumor cells, are not relevant as measured by immunohistochemistry classically in terms of predicting. So intuitively you would have thought, well, you having a high expression, you could do better because at the end of the day you will accumulate more chemotherapy payload from the ADC. But for most of the cases, this has not been the case. So one of the issues is that quantitative assays with immunohistochemistry are very often flu, so they are not really quantitative. So is that going to be a way to help us to really get more quantitative assessments using artificial intelligence? And the second question is, how are you going to validate those findings? Because the issue is that you generate a measurement with artificial intelligence, which is based in, I don't know how many parameters, and actually, I don't know which parameters. So then how are you going to validate that? So I'm thinking like, instead of getting 100 parameters or 1,000 parameters from pathology, artificial pathology, you could think about taking into account the color of the eyes, the color of the hair, the color of the socks, and whatever, you could be able to identify on a cohort, define an algorithm to select patients. The problem would be to apply this algorithm to a second data set and see that this is somehow confirming that it's working. So, I mean, how are you taking that? I mean, the good example is, this data presented with the TROP2 membrane cytoplasm ratio. Are you really a believer on that, or how do you interpret those data?
I mean, regarding the possibility of using AI to predict which patients are going to benefit from antibody-drug conjugates using IC stains, yes, Luis, I'm super convinced that this is going to be really helpful, because really the human eye is not able to really determine particularly intensity. And I think here we are talking about intensity of staining. Also, we are not very good when we have large sections to score things. And so this is what we have traditionally used with the H-index. So I think with the ADCs, this is going to be really helpful. But I share your concerns regarding the validation of all these tools. That's why I always think the validation is not the comparison with humans, but the comparison with clinical outcomes. I think that's going to be easier than in the past, because really here you don't need to move samples. You just need to upload images to a server, so many independent groups can work with the samples. And I have seen more collaboration here in the AI space than in other previous projects, for example. But to your point, some of the things we are doing now, we are using 5 cohorts to validate. We're using an initial discovery cohort, first a validation cohort, then 2 cohorts from clinical trials, and maybe a final cohort from clinical practice, because there could be a lot of bias. I fully agree.
Okay, so I think we can go to the next speaker, and really happy and really delighted to have here Professor Julian Dongwer. He's coming from the Crick Institute in London, and he's going to tell us about investigating the interplay between KRAS oncogenic signaling and tumor immune evasion in lung.
Thank you very much, and thank you for inviting me here to Madrid. It's always a great pleasure to come to Madrid and to discuss with you work that we've been doing on lung cancer, in particular RAS signaling in lung cancer. So what I'm going to talk about today, I will, by way of introduction, talk about the challenge of curing KRAS mutant cancers by targeting RAS oncogenic signaling and the problems with resistance and how this is really limiting the efficacy of drugs that have been developed recently targeting KRAS. And I'll go on to talk about combination therapy approaches that we're taking, principally using preclinical models, mouse models, where we look at targeting RAS and at the same time targeting what's going on in the tumor immune microenvironment, so the components of the immune system that are coming into the tumors, to see how we could increase the impact of inhibiting RAS and to reduce resistance or delay resistance. And finally, more briefly, I'll talk about some more speculative work we're doing, looking at the possibility of using the killing of the drug-sensitive tumor cells, RAS-bearing drug-sensitive tumor cells, to prime an immune response against cells within the same tumor that are becoming drug-resistant, as a way of perhaps thinking slightly differently about this drug resistance problem. So, as I'm sure everyone is aware, the RAS oncogenes are a major clinical problem, probably in the order of 2 million deaths a year globally caused by mutant RAS oncogene-driven cancers. And the focus for my lab has, in recent times, has mostly been in the lung, where KRAS mutations are pretty frequent, and in particular, you very commonly get this KRAS G12C mutation, which is not seen so much in other tissues, but is a classic mutation caused by the carcinogens in tobacco smoke, benzoate-pyrene in particular. And this has led to sort of first generation of drugs targeting RAS, which I'll come to in a moment. So what RAS is actually doing as a protein, just by way of sort of deep background, is it's a GTP binding protein, small GTP binding protein, cycling between an active GTP bound state and an inactive GDP bound state. And it's getting signals from many upstream signaling components, particularly receptors and receptor tyrosine kinases and other types of receptors. And this is pushing it into the activated GTP bound state. And it's then hydrolyzing GTP and becoming inactivated. And this hydrolysis reaction is promoted by GTP binding proteins, and those are also lost in some tumors, particularly NF1. And the oncogenic mutations you see in RAS are actually blocking the hydrolysis reaction, so why the RAS ends up in this activated state. And so RAS proteins sit in this signaling network, controlling a lot of downstream targets, most particularly the RAF-MAP kinase pathway, and there are a lot of complexities to these pathways, which I can't go into here, but suffice to say that they've been the subject of a huge amount of drug discovery efforts, and actually all these flashes here are drugs that have either been approved or in clinical trials. So it's a really central driver of growth in cells, but also many other things, survival and metabolism and all manner of important things that cells do. And in the last decade, we've started to make progress against targeting RAS itself and not just other things in the pathways. RAS was a very difficult protein to target for a number of reasons, but it was finally cracked, or at least the initial pathway to cracking the targeting RAS was made by Kevin Shokat when he used this neomorphic cysteine residue that is in the KRAS G12C mutant protein to covalently bind inhibitors that would then lock the RAS protein in its inactive state. And that's led to a number of drugs, two of which have been approved for clinical use, sotorasib and adagrasib, which target that specific mutation. And I would say, and there are a lot of other ones now as well that that have been developed, a lot of inhibitors of other isoforms of RAS, the other mutant forms of RAS, G12D in particular, and also pan-RAS inhibitors and KRAS isoform selective inhibitors. I would say there's an advantage to the mutant selectivity of the first generation of inhibitors being developed, because obviously RAS proteins have a lot of important normal functions in cells. And actually one of the first sort of, an early piece of work that I was involved with on RAS showed that when you activate T-cell receptors, you get this very rapid activation of RAS within a few minutes, and you can see the RAS being pushed from a GDP-bound inactive state to a GTP-bound active state when you stimulate the T-cell receptor within about 10 minutes. So if you have pan-RAS inhibitors, the likelihood is you'll interfere with these sorts of things. I mean, obviously, there could be therapeutic windows depending on how sensitive different cell types are, but this is clearly a problem, and I think this has led to concerns about pan-RAS inhibitors, but that doesn't apply to mutant selective inhibitors. So there's been this huge explosion in the development of inhibitors against RAS, and here are some of the ones that are in trials. It's very hard to keep up with progress in this. This was, I think, sort of the state of play early this year. And you see the ones that are approved, sotorasib and adagrasib, and then a whole lot of other ones that are targeting the G12C off state, but then also some on-state inhibitors from Revolution Medicines down the bottom left there, and a whole lot against G12D and other variants. So there's been enormous progress in making RAS inhibitors. The issue is how well do they work, and so sotorasib and adagrasib, the ones that are approved, and there's a lot of clinical data on them now, particularly on sotorasib, which was the first to clinic. And so you can see here from the phase 2 trial, which it was actually approved on an accelerated FDA approval, you can see the waterfall plots look very good for sotorasib. This is in G12C mutant non-small cell lung cancer. But the impact is pretty transient, so you get this rapid development of resistance, and this is the progression-free survival from the phase 3 trial that followed up on the after the approval. And so actually the progression-free survival benefit is only 4 to 6 weeks, which is not a lot. And actually when you come to the overall survival, that trial failed to find any benefit to overall survival through the use of sotorasib, which I think for me, and I'm sure for Mariano, people who've worked on RAS for 4 decades or so, this is not really where we felt we would be once we had good inhibitors of these drugs, of these targets. I mean, I guess there is an issue as to how good these drugs are, but also just how much traction we can get out of targeting this specific target, KRAS, in a real disease setting. Obviously, very rapidly, people have looked at mechanisms of resistance to RAS inhibitors, and unlike in the case of EGF receptor inhibitors, where there is a predominant mechanism of resistance and then some other sort of more minor secondary mechanisms, in the case of the RAS inhibitors, the resistance mechanisms are really all over the place. Secondary mutations in RAS are the ones that make a different way of activating the other allele of RAS, or that prevent drug binding, activating mutations downstream in the MAP kinase pathway, upstream in the receptor tyrosine kinases, or overexpression of receptor tyrosine kinases, or overexpression of ligands for them, and then also orthogonal pathways such as the YAP pathway, and changes in cell histological state, so the small cell lung cancer transformation and epithelial mesenchymal transition, which always appears in drug resistance mechanisms. So really all sorts of things happening, and from this it's very hard to sort of see how you could take an approach of trying to suppress these other resistance mechanisms in a useful way. So the clinical impact of RAS inhibitory drugs to date has been pretty modest, and actually we didn't mention many patients actually have pre-existing resistance. So it's only about half of the patients really give a significant response to these RAS inhibitors, and those that do respond, you get quite a rapid onset of resistance through highly diverse mechanisms. So I think to really make an impact in the clinic with these drugs, tackling this problem of drug resistance is going to be really central. So how can we increase the clinical impact of RAS inhibitors? And so one of the areas that we've been following up on here is to look at combination approaches. So what other things can we combine the RAS inhibitors with to try to increase their impact? And there are sort of 2 main ways one can do this. One is to look at tumor cell intrinsic combinations. So you think of the tumor cell and you want to increase the depth of the pathway inhibition. And so I think I've got I do have a pointer, yeah. So with RAS, you're inhibiting the off state here with drugs like sotorasib, but you can also add on inhibition upstream or downstream, which may increase the depth to which that inhibition goes. And another way is to try to do things that are completely orthogonal and within the tumor microenvironment. So one of the things that RAS inhibitors do one of the things that RAS does is to promote a tumor immune suppressive microenvironment. So they drive a transcriptional program that produces a lot of cytokines that are immunosuppressive. And so when you treat tumors that have RAS mutations with RAS inhibitors, you tend to, for a period of time at least, that immunosuppressive microenvironment is reduced or degraded to some extent. So there may be things that one could do to try and boost the ability of the immune system to get into the tumors at that point when they're more vulnerable. So RAS is controlling the tumor immune microenvironment in a lot of different ways. It's a very complex set of things that are going on here, but essentially all these different components of the tumor microenvironment are being impacted by RAS signaling in one way or another. And I particularly point out the production of cytokines like ones controlling macrophages, such as the IL-6 and GM-CSF, producing a sort of a immune suppressive macrophage, bringing immune suppressive macrophages. Ones like IL-10, TGF-beta that are bringing in regulatory T cells that I'll talk about more later. And then there's also a number of other mechanisms like adenosine production, which is interstitial adenosine, which is immunosuppressive, lactate production, which is also immunosuppressive. And also critically inhibiting the interferon response over on the right-hand side there. So all of these things are RAS is controlling, and if you inhibit RAS, then these signals get reversed, at least for a time, and there may be an opportunity to promote immune attack during that period. So the RAS inhibitors promoting this sort of more immune surveillance response. And you can see the difference that this makes in mouse models. Just by way of illustration here, I'll talk more about these models in a moment, but this is an immunogenic lung cancer model, mouse lung cancer model that is KRAS mutant p53 deleted. And if you treat it just for a relatively short time, 2 weeks, the gray blocked out area is when they're being treated. And in this case, they're being treated by a sort of a next generation RAS inhibitor from Revolution Medicines that targets the activated form of KRAS G12C. But the graphs wouldn't be that different with sotorasib or adagrasib. And what you can see in the immune competent mice, actually just with this, because this is quite an immunogenic tumor, just those RAS inhibitors in a subcutaneous setting can cause complete cures of those mice. Now that's probably not particularly relevant to what you'd see in a patient setting, but at least it sort of shows that in a favorable setting, the immune system can, you can get complete eradication of these tumors. And if you look on the far side there is what happens if you do that in a rag null mouse, and you see there is no eradication of those tumors. So that ability to not just cause the tumors to regress, but to actually eradicate the tumors, that's entirely dependent on the action of the immune system on those KRAS mutant cells. So we use a number of mouse models to look at this problem, to model it in mice, and really they divide into three main areas which sort of also correlate with the what you see in the clinic. There's up the top there we call the immune desert model, where there's really no engagement of the immune system at all, and those tumors typically have very low antigen neoantigens. That's less common in the clinic. That some of the EGFR mutant cancers are like that. In the middle, there's probably the most common clinic, these immune excluded ones, which are also cold in that they don't have affected immune cells in them, but they have a lot of myeloid cells in them, and particularly a lot of immunosuppressive cells. So there's clearly an interaction with the immune system going on there, but the immune system's been completely shut down locally in the tumor. And then the bottom, this is the one that I showed you the data on just now. This is an immune inflamed model where you have T cell infiltration, and these are the ones that will respond to immunotherapy, both in the mouse models and clinically. And the mouse models we use, we're using syngeneic transplantation models. This top one is just derived from a KRAS P53 mutant GEM, genetically engineered mouse model, and there's nothing else has been done, but that has very low mutation burden. This middle one is from an old Lewis lung carcinoma model, and these are very immunosuppressive because they've actually been passaged through immune-competent mice. So they've really got a lot of immunosuppressive mechanisms operating in them they've been selected for. We call them delta NRAS because the three alleles, this Lewis lung carcinoma derivative has a naturally occurring KRAS G12C mutation, but interestingly, it also has an NRAS mutation. And in order to make it sensitive to the KRAS G12C inhibitors, we deleted the NRAS by CRISPR. And then the bottom one is one that we've engineered, again, from a genetically engineered mouse model where we had attempted to increase the tumor mutational burden by expressing a cytidine deaminase in the tumors. And actually, there's a long and complicated story about those. This cell line in these mice. Turns out that the reason that they're sensitive to immunotherapy is not due to the mutations introduced by the cyclin deaminase, but actually due to expression of an endogenous retroviral antigen, an envelope protein. And then we had a sort of whole story about that in Nature last year, where we showed that that was giving a strong B-cell response, and that's also relevant in quite a lot of tumors in the clinic as well, in lung adenocarcinomas that show this B-cell response to endogenous retroviral antigens. So when we treat these immune hot models with immunotherapy, they are slightly sensitive to immunotherapy. That these, so I'm not doing very well with this pointer. So there's a weak response, that's that orange line, to the immunotherapy, but it's always consistently there. And the response to the RAS inhibitors, the green line, is reasonable. They're only treated for 3 weeks here. But if you combine the immunotherapy with the RAS inhibitor, you start to see these combination responses. This is an orthotopic setting, which is always more difficult to treat. And if you look at the actual tumors, this is just a fact showing the expression of T-cell exhaustion markers on T-cells within the tumors. And on the right-hand side, that's the immune hot model, showing that T-cells are coming into those tumors and engaging, being engaged and going through the activation and exhaustion cycle. Whereas on the one on the left, KPV6, that's the immune desert model where there's no neoantigens, there's really no engagement with the immune system at all, no sign of any T-cells that are in those tumors that they've been in any way aware of any sort of stimulus. And when we look at the immune cold excluded model, the 3LL model, we see that there is also no impact of a PD-1 therapy there, just shown in terms of the size of the tumors, again in an orthotopic setting. So those cold models don't respond to immunotherapy. They don't respond to a combination of immunotherapy with the RAS inhibitors. And if we look in what's going on in those tumors and all those different models, we see that that when you add the RAS inhibitors to them, we see a series of things going on that are fairly similar between the models. That you see this reduced immunosuppressive cytokines and less myeloid cell infiltration. That's particularly important in the immune-excluded models. We see some signs of tumor cell death, dendritic cell activation going on, and T-cell chemotaxis coming into the tumors, also antigen presentation and T-cell activation and exhaustion. We also see inhibition of the MYC pathway, as you might expect, and this seems to be linked to the increase in interferon response. So these various analyses described in these papers here that we published over the last couple of years. So although in the different models you're seeing fairly similar things happening, which you'd all expect to increase the sensitivity to immune checkpoint blockade, it's actually only in the immunogenic models that actually makes a difference, and you actually start to see responses to that combination. In the other models, you don't get above some sort of threshold to actually get an impact of the immune checkpoint. So you're getting all these effects of the RAS inhibitors on the immune system, but only in cells in tumors that are already immunologically hot does this actually reach a threshold for that immune checkpoint blockade response, or any sort of combination responses. So to try to see how we could improve on this by adding other things into the combinations that we're using with the RAS inhibitors, we've looked at a number of things, and I'll tell you today about the one that's sort of been most rewarding, which is the SHP2 inhibitors. So SHP2 is a tyrosine phosphatase, which also acts as an adapt protein in a number of signaling pathways, including the RAS signaling pathway. So actually in tumor cells, when you add SHP2 inhibitors, you will increase the depth of inhibition of the RAS pathway when you have it together with a RAS inhibitor. And this is particularly true for the RAS off-state inhibitors, but actually also you see it with the on-state inhibitors as well. And so you can see this RM29, that's the RevMed RAS G12C on-state inhibitor. And you can see that after 48 hours of treatment, you're starting to get reactivation of the ERK pathway. The SHP2 inhibitors do also slightly inhibit the pathway, but not as well as the RAS inhibitors. But the combination of the two of them actually gives you much more durable inhibition of that pathway. So you're probably getting some benefit there. But probably more critically with the SHP2 inhibitors, you're having this effect on the tumor microenvironment. And the most predominant thing that's seen is really this repolarization of the M1 type macrophages, which are targeting tumors, to the M2 type the inhibitors cause a shift from the M2, which is immunosuppressive, to the M1, which are targeting the tumors. And there was this paper from Revolution Medicines in 2020 described that change. So in the tumor cells, SHP2's sitting upstream of RAS, and so inhibitors of that promote the depth of inhibition of the RAS pathway. But then in the immune microenvironment, or in the microenvironment generally, there are all sorts of places where SHP2 is acting, and generally acting to restrain the immune response. So the inhibitors are actually promoting immune response. They also promote normalization of the vasculature as well, which also helps. So if we have a combination of the RAS inhibitor, in this case we're using this another RevMed drug, well actually they just changed the name of it, RMC-4998 is the G12C RevMed drug, and their SHP2 inhibitor, the 4550, we can get, even with just 2 weeks of treatment in these subcutaneous immune hot tumors, we can get complete eradication of those tumors, and that's a sort of stronger response we get with just the RAS inhibitor by itself. And if we do it in the Ragnall's mice, we see we don't get that at all. So there's all an immune-mediated promotion of that destruction of the tumor. And you can see that in the orthotopic setting as well, and this actually shows both the impact of combining the RAS inhibitor with the SHP2 inhibitor, and also with PD-1 inhibitor. So the PD-1 inhibitor is actually a good combination there. One of those top two lines is RAS plus PD-1 inhibitor. So these combinations of SHP2 inhibitors and PD-1 inhibitors with the RAS inhibitors can actually give you elicit immune-mediated cures in these preclinical models with the immune hot tumor microenvironment. And this is an area that's obviously been very interesting in clinical trials, and people have been particularly combining RAS inhibitors with PD-1 inhibitors, but have run into a lot of toxicities. And I think the hope is that this will get better with more specific drugs, and I think the indications are that is the case. But it's certainly the case that sotorasib and probably adagrasib combine very poorly with immune checkpoint blockade, particularly with problems of immune-mediated hepatotoxicity, which were really very bad in those combination trials. But I think the indications are that that's a particular problem with those drugs, and obviously these covalent drugs do tend to have quite high toxicities, at least in the first iteration of them, and they need a lot of engineering to make sure that they're highly specific and you're not getting off-target binding. And actually an interesting study was done on, if you take a mouse and you treat it with sotorasib and then you do mass spec on it, where does the drug end up? And actually it's, there's a little bit of it in the tumor, but the vast majority of it is covalently bound to proteins like hemoglobin and tropomyosin and all these things. So you can see why that could cause significant toxicity problems. So I think it's clear that in the immune hot setting, you're going to get pretty good combinations of responses between RAS and immune checkpoint blockade if you can sort out these problems with the toxicities, which I think will happen. I think what's going to be much more problematic is in the immune cold setting, And I would say also something to bear in mind with all the clinical trials that have been done, the vast majority of the trials combinations, patients have already failed on immune checkpoint blockade. So they are already, pretty much by definition, they're already immune cold going into those trials. So from all the preclinical data, we would expect it to be unlikely that you would see a combination response to the immune checkpoint blockade and the RAS inhibitors in patients that have already failed on immune checkpoint blockade. So we've been trying to sort of understand what's going on in the immune-excluded tumors and how this might be improved upon. And we've taken a number of approaches with this, and I'll tell you some stuff about some highly multiplex spatial approaches as well that have tried to look in more detail at what's going on in the tumor microenvironment. So this is in subcutaneous setting, these immune-excluded tumors, and we're seeing the treatment over a short period of time here, shown 2 weeks, what you get with a RAS on inhibitor and a SHP2 inhibitor. And so you're seeing some reasonably good combination approaches, but combination effects. But as you carry on with treatment, and now, unlike in the immune hot models where we were just doing short periods of treatment and then looking at what happens after you withdraw the treatment. Now in this immune excluded setting, we keep the treatment there the whole time. And so you see even on treatment, these drugs stop working after about 3 weeks. You start to see the resistant disease come up. And if you go in and you look at what's going on in those tumors, you can see that there are clearly benefits being had from having the RAS inhibitor and the G12 and the SHP inhibitors together. And although I can't use a pointer, if you look at particularly on the right-hand side of these blocks, you can see the increased levels of CD8 T cells and NK cells and dendritic cells that are coming with the combinations that are better than with the single agents. And then down here, the immune suppressive drugs, the immunosuppressive cells like these M2 polarized macrophages are very much being depleted by these therapies. And this is a 6-day treatment. So these therapies are sort of tipping this balance towards a more immune targeting tumor microenvironment. And so can you actually sort of push it over so you actually start to get responses that are lasting. And I should say that, so you get this increase in T cells in the tumors. So the right-hand panel, right-hand block for each of these is the combinations. So you're getting more and more CD8 T cells coming in and CD4 helper T cells. But also, I'd point out, you're also getting these regulatory T cells coming along, which are probably coming along as a result of following the immune effector T cells. And that's a problem that we'll come to in a minute. In terms of the response to therapy, so here is, so this is the combination up here of the SHP2 inhibitor and the RAS inhibitor treated for 30 days, and you see the tumors are all getting out of control. But if we add in checkpoint inhibitors, we're starting to get some sort of control of these tumors. This is adding PD-1 into the mix. And but that's despite the fact that the immune checkpoint inhibitors by themselves, either PD-1 or CTLA-4 or a combination of them, aren't doing anything at all to these tumors. They by themselves or the two together, they can't have any impact at all. And actually, when we use the combination of the PD-1 and the CTLA-4 with the SHP2 inhibitors and the RAS inhibitors, we can actually get complete eradication of these tumors every time, and with the establishment of immune memory, so those mice are then resistant to re-challenge. So it does show that actually, there's a finite number of ways in which these tumors are avoiding the immune system, and by these approaches, you can actually block all those off. Now, obviously, a four-way combination of drugs like this, several of which have significant toxicity problems, is unlikely to be something that one could take forward into clinical use. But I think it does establish the principle that however immune-evasive the tumors are, there is the potential to eradicate them through the use of the immune system in combination with the RAS inhibitory drugs. But we may need to find less toxic ways of doing that. So, as I mentioned, we've done a multiplexed spatial biology approach to looking at these tumors. So really looking, actually using imaging mass cytometry to look at a lot of markers, 36 markers in these tumors when we're treating them with these RAS inhibitors. And this is an orthotopic setting shown here. So this is one of these 3LL tumors, the cold ones that are immune excluded. And you can see that there's very small numbers of CD4 or CD8 T cells within the tumors. But when you treat with the RAS inhibitors, you see these cells coming into the tumors. So particularly you see CD8s coming into the tumors. So clearly there is, you're building on the potential to get an immune attack on the tumor. But the RAS inhibitors by themselves won't sort of push that over a threshold where there's actually an effective response. So using this imaging mass cytometry analysis, and so to say, this was actually, we published, so it's falling off the bottom there, we published an initial analysis this a couple of years ago in Nature Communications, but we have a paper just coming out in Science Advances where we've taken this further, and particularly on the sort of the analysis side, and trying to understand all the juxtapositions of the different cells within the tumor microenvironment. And although I won't have time to go to this in detail, the paper is on bioRxiv if you're interested. One can see that what Megan Cole, my graduate student, did here was she analyzed, she identified all the different cell types within the tumor, and then she did this neighborhood clustering process where you look at what each cell is surrounded by, what sort of cells surround each cells, and then you can classify each area into communities. So you're saying that this is an area where there is a certain ratio of T cells to dendritic cells to tumor cells, this type of approach. And you can do it in many different ways, but the way she analyzed it, she ended up with these 18 different communities within the tumor microenvironment, and then was looking at how those would change on treatment with the RAS inhibitors. And so the communities, there are ones that are in the normal tissue, the ones that are at the interface of the tumor and the normal tissue, and then the ones that are in the tumor itself. And these change quite radically when you treat with the RAS inhibitors. This is the Mirati, their tool compound for adagrasib. So you see this big sort of switch between these communities, reflecting this reorganization of the tumor microenvironment when you treat with the RAS inhibitors. And we particularly followed up on the 5 communities that had the highest CD8 positive T cell count as being the ones that we thought might be most important in getting an immune response against the tumor. And some of these go up a lot when you treat with the RAS inhibitor. And you see them also moving into the tumor core. So on the right-hand side there, you can see there are 3 communities, particularly that build up to quite a big extent. This is after 6 days of treatment within the core of the tumor, and those are the ones we were particularly interested in. And you can see them represented here on the actual, on the tissue sections. And there was one particular one of those which we call community TDC, which has got a lot of T cells and a lot of dendritic cells. And the particularly interesting thing with that was a few features. One was that the T cells, the CD8s and the CD4s, were all proliferating in that community, as were also regulatory T cells within that community. And also, very interestingly, the T cells in that community were all, following treatment with the RAS inhibitors, were associating, many of them were associating with caspase-3 positive tumor cells, so indicating that there was actually killing going on of these tumor cells by the CD8 T cells, specifically in that community. And this community also, interestingly, was one of the ones that had a very high, where most of the regulatory T cells were. There's one other that had a sort of similar amount of regulatory T cells. But this seemed to be a community that we were particularly interested in, as it looked like there was interesting stuff going on with tumor cell killing, but there was also all these regulatory T cells. And if we looked at the distribution of how close the CD8 T cells are in that community to dendritic cells and also tumor cells, we see that induced by the RAS inhibitors. Those sort of blocks on the right show that. But there was also signs that regulatory T cells were also close to the CD8 T cells. And you could subdivide that community, and some of those communities didn't have regulatory T cells in, some of them did. And the ones that did have regulatory T cells, you saw this very strong effect of the proximity of the CD8 T cells and the dendritic cells and the tumor cells induced by the RAS inhibitors. But in the communities which were otherwise the same type of community but just lacked the regulatory T cells, that had the regulatory T cells, the upper one is one where they didn't have the regulatory T cells, you really suppress that effect. So it looked, sorry, I didn't explain that very well, but it looked as though you were getting a situation where in that community, if you had regulatory T cells there, they were shutting down this CD8 response, whereas if the regulatory T cells weren't there, the T cells, the CD8 T cells, were engaging the tumor cells and also with dendritic cells. So this made us think that actually this regulatory T cell presence could be very important in disrupting the impact of these CD8 positive T cells that is induced by the RAS inhibitors. So regulatory T cells are one of the things that CTLA-4 targets. So it's extraordinary that after ipilimumab has been in the clinic for well over a decade, there's still a lot of argument about how it works. And one of the things it does is target regulatory T cells, but it also does a number of other things. And but it may be that if the regulatory T cell targeting is the key thing, that one could do it in a way that is less toxic than the effects that have sort of plagued ipilimumab. And so there's been some studies done where people have tried to find what's special about Tregs in the tumor versus elsewhere. So it's well known that you need Tregs. If you make a mouse without Tregs, you will rapidly get a horrible autoimmune disease. So you can't just take out Tregs. But there's something special about the Tregs in the tumors that would allow you to have some differential there. And so a couple of studies done a number of years ago looked at basically looking at expression of genes in regulatory T cells in the tumors versus in normal tissue. And they both came up with this molecule CCR8 as being selectively expressed in tumor-infiltrating Tregs. So there's been a lot of interest in targeting CCR8 as a way of targeting Tregs. And we could look at this in our model system and combinations of the RAS inhibitors and PD-1, and then either CCR8 or CTLA-4. And you can see actually the best response over there is using the CCR8 antibody. So maybe that CCR8 is actually able to do this rather better than CTLA-4 targeting. And if you look at what's actually going on in the tumor microenvironment there, you can see here on the left, the right-hand two bars there show how the Tregs are actually depleted by that CCR8 antibody. So it's a depleting antibody that one uses that's actually causing removal of the Tregs. And that this also further promotes the CD8 T cell influx into the tumors. So combination targeting Tregs along with KRAS and SHP2 may be an effective way of basically sort of turning these immune-evasive tumors from a cold state to a hot state and making them responsive to these these combinations of immunotherapy. So I think that's an area that we're very interested in sort of following through in clinical setting. So finally, I'd just like to sort of talk about some more speculative things about how we might be able to promote the immune response against tumor cells as a way of targeting drug-resistant cells within a tumor. So can we use the immune response against the drug-sensitive cells to set up the immune response against the drug-resistant cells, because clearly the drug-resistant cells and the drug-sensitive cells are all very closely related and share antigens. So to do this, we designed a model system. This is my graduate student, Mona Tomashko. And this, again, we're using this immune hot model that we call KPAR. And she took this, and we'd made different versions of this, one of which has KRAS G12C, and another has KRAS G12D. So the G12D version will not be responsive to the G12C inhibitors, whereas the G12C version will be responsive to the G12C inhibitors. And yeah, as I mentioned before, this model is actually sensitive because it has this, it's re-expressing this endogenous retrovirus envelope protein, which, as I said, you do also often frequently see in human clinical cases as well. So Mona labeled these up with different fluorescent labels and also a luciferase label, and there are also barcodes in these labeled cells. And this allows one to trace these cell populations. And in order to avoid any immune responses against the labels, we did this in a mouse called glowing head mouse, which expresses these fluorescent proteins and luciferase in the pituitary glands. So they have, if you look at them by a luciferase in a bioluminescence scanner, which we used looking at the tumor size, their heads will be glowing. So they're called glowing head mice. But it means they're tolerant to these labels. And so we can see the tumors by luminescence, and we can also image them with antibodies. So in this case, GFP and BFP are almost identical sequences, 2 amino acid differences. So the GFP antibodies will recognize all the tumor cells, whereas the luciferase antibodies will recognize only the G12D ones. And then we can also pick them up, obviously, in the FACS as well. And so if you look at so Mona went through a lot of iterations of what would be a good balance of resistant cells and sensitive cells to use to model what goes on in a tumor when you have a minor population of drug-resistant cells that expands up on therapy. And she came up with this ratio of putting in 0.04% of the drug-resistant cells. And this work she's principally been doing in subcutaneous tumors. There is a little bit of off-topic work as well. But so this level, you've got a tumor established, and you're putting in, at the beginning, there's about 100 drug-resistant cells in there, which are just scattered in the tumor. So you treat them with the RAS inhibitors. And then if you look at the tumor bulk with this Revolution Medicines RAS inhibitor, you see the tumors regress, start to regress, but then after 2 weeks on therapy, they're already beginning to come back. Now, if you hadn't had the drug-resistant cells in there, those tumors would stay down for as long as you keep the therapy on, because they are very sensitive to the therapy. And then if you look at the bottom of the luciferase, which is marking the drug-resistant cells, you can see that in the vehicle-treated mice, you really don't see the luciferase coming through at all. Those cells are a tiny fraction. But in the case of once you've treated with the RAS inhibitors, then you start to see them take off, and they will grow up quickly. And at the end of the treatment period, if you take those tumors and you FACS sort them, you see that in the vehicle-treated, there's basically effectively no drug-resistant cells, or such a small population they don't register. But in the RAS inhibitor treated, it's basically 100% are those drug-resistant cells, so all emanating for that 100 cells that you put in at the beginning. And so you can look at what's actually sort of happening in those tumors. So if after 6 days of therapy you cull the mice and stain the tumors, you can see, so here the GFP staining shows you all the tumor cells within the tumor on the top there. And then the luciferase staining in yellow here shows you the drug-resistant cells. So the drug-resistant cells are expanding up in these sort of nests of luciferase-positive drug-resistant cells. And if you go in, look at those more closely, and for example, do a Ki67 stain, you can see that not surprisingly, that the drug-resistant cells are all staining well for Ki67. They're all proliferating, whereas in an area where there aren't any drug-resistant cells, there's very little proliferation going on. And you can take those cells out and look at them, look at what they're expressing, for example, so you can fact sort them and then do RNA sequencing. In this case, to show you some bulk RNA-seq, and we're doing this with single cell as well now, but with bulk RNA-seq, you can see various things happening. So we have here is we're actually using either the G12C inhibitor or the SHP2 inhibitor in the treatments, or a combination. And the G12C cells are on the left and the G12D cells on the right. And so what you see with the G12C cells is that you see all these cell proliferation targets going down. So these cells are stopping growing. Whereas on the resistant cells on the left, up at the top there, they're actually going up. So they're benefiting from the lack of competition, and they're starting to go up. What you see next down from there, in the red box, is the interferon responses, which go shooting up in the drug-sensitive cells. So this is a classic thing that happens when you treat with the RAS inhibitors. The interferon response goes way up. And interesting, on the drug-resistant cells, although they're sort of growing in these nests, you're seeing signs that actually those are are also showing, not as much, but they are also showing this interferon response. So clearly they're getting a knock-on effect from what's happening in the drug-sensitive cells, where this interferon response has been triggered. That's impacting on the drug-resistant cells as well. So in this model, the cells that we use are actually, the drug-resistant cells are not responsive to the SHP2 inhibitors by themselves. So this is sort of a quirk of RAS biology. So the SHP2 inhibitors will have an effect on G12C tumor cells, tumor cell autonomous effect. But because of the way RAS cycles and the difference between a G12D and a G12C mutation, it won't have an effect on the G12D tumors. So the SHP2 inhibitors actually won't inhibit them as a single population, which is just a single all G12C or all G12D on those two sets of mice shown there. And also, actually, the G12Ds are rather less sensitive to the PD-1, not that either of them are that sensitive. But so neither the SHP2 nor the PD-1 is inhibiting the drug-resistant cells when they're by themselves. But if we look at the combination done on these mixed tumor populations, we can see, so again, the RAS inhibitor is having this sort of transient effect, but over towards the right there, when you have the RAS inhibitor plus the SHP2 inhibitor, you see that effect is no longer transient, and actually you're really taking out those cells. And if you look at the bottom, you can see the drug-resistant cells through the luciferase readout, and those are also being impacted. So that's despite the fact that neither SHP2 inhibitor nor the RAS inhibitor is affecting those G12D cells by themselves if you grew them as a homogeneous tumor by themselves. But when you're growing them in a mixed tumor, then they're being impacted by those combinations. And it's actually the same story with the PD-1 combinations there as well. And you can get, if you look further out, you can start to get complete responses. So with the combination of the RAS and the SHP2 inhibitor, you can get, if you're following it out for 2 months, you can get about half the mice are cured there. And similar things with the PD-1 combination. And the three together, you're getting most of the mice being cured. And if you re-challenge those mice, then many of those have got immune memory. And that effect is entirely dependent on the adaptive immune system. So if you do it in wild-type mice, you see it, 7 out of 14 complete responders there with the SHP2 RAS inhibitor combination. But if you do it in the ragdoll mice, then no complete responders no eradication of the resistant population within the predominantly sensitive tumor. Going in and just looking at what's happening in the tumor microenvironment there, as we've seen in other cases, you can see how profoundly, in the bottom right there, how profoundly you're getting this influx of T cells, CD4s and CD8s, into the tumors when you've got these combinations of RAS inhibitors and SHP2 inhibitors and PD-1 inhibitors. So, to conclude that section, we think that the adaptive immunity is contributing to the efficacy of the G12C inhibitors. I think that's clear in all the mouse models we look at. But if we have these mixed models where we're putting in resistant cells, if you just use the RAS inhibitors, clearly those resistant cells gain a proliferative advantage, as you'd expect that you're getting rid of the competition. And if there is some knock-on effect, as we've seen in the RNA sequencing, that they're starting to see an interferon response, it's not enough to really slow them down significantly. But when you start adding in other things like SHP2 inhibitors and the PD-1 inhibitors, you can actually get complete responses there. So you can get, essentially what's happening there is there's an immune-mediated bystander killing. You're targeting sensitive cells, and this is triggering an immune response against cells that are not responsive to that drug combination directly. So, and that response is one that we're really interested in, this bystander immune-mediated killing, and we're trying to sort of untangle that further now by going in and really studying what it is that's killing those cells and what they're responding to. So I'll finish there and just say that RAS inhibition remodels lung tumor microenvironment, and this is giving us a lot of opportunities for combination with immunotherapy. And I think that where we are now with RAS inhibitors in the clinic, it's clear that however good those RAS inhibitors are, and I think the first generation ones aren't very good, but however good they become, probably the best one could hope for is probably a lot less than we see with osimertinib in an EGF receptor mutant lung cancer, because the nature of these RAS mutant lung cancers, they're much more mutated. They have a much higher tumor mutation burden. They're going to have resistance mechanisms that are going to come up quickly, even if the RAS drugs are absolutely perfect. So we've got to tackle this issue of how we combine them with other things. And I think there's so much going on with RAS in the immune system that there are many possible avenues. And what I've talked about today is combining with SHP2 inhibitors, combining with PD-1, PD-L1 inhibitors, and approaches that use depletion of Tregs to promote the immune response when you inhibit RAS. And I'm hoping that one way or another, with all of these different things, we may be able to start getting some real beneficial combination responses between RAS inhibitors and various types of immunotherapies in the future. So I'll stop there and thank people in my lab. And in particular, I'd like to highlight Miriam Molina, who really co-supervises most of the work in the lab, Megan Cole, who did that work on the imaging mass cytometry and the analysis of that, and Panos Anastasiou, who did the work on the SHP2 combinations, and Mona Tomashko, who's done the work on the drug-resistant subpopulations. And thanks to all the funders there. So thank you very much.
Hi. I was wondering about the immune infiltration that you're promoting by inhibiting SHP2 and KRAS. Is that irreversible? So if you remove the treatment, the immune cells remain inside the tumor, or?
No, that reverses. So if you remove the RAS inhibitors within a few days, the immune suppressive tumor microenvironments have been reestablished. So you see the T cells excluded again, and the myeloid cells come in. Yeah, so it turns around quite quickly. We've certainly looked at 4 days, and it's pretty much reestablished then.
Because that I was asking because So the timing of the potential treatment that might arise from here, that's very relevant, no? So would you give the PDL inhibitors at the same time as the other inhibitors, or it would be sequential, or what would be the best?
I think, you know, sort of, aside, not sort of thinking of any of the sort of possible toxicity issues, but from just the point of view of what we think in terms of promoting efficacy, I think you'd ideally want to have the immunotherapy there at the point that you start the RAS inhibition. I mean, maybe a little bit before, but it should all be inhibited by the time those RAS inhibitors hit the tumor. Yeah, I think.
Okay, thank you.
Great talk, Julian. I have a problem with the mechanisms of resistance. Everybody has identified novel mutations, okay? But I don't have clear in my mind what is the contribution of those novel mutations to the resistance. For instance, if you have a resistance where BRAF is mutated, I haven't seen anybody combining the KRAS inhibitor plus the BRAF inhibitor and eliminate the resistance. Let me just, I have the suspicion, and I want to hear your opinion, that the main mechanism of resistance is KRAS amplification. I don't know whether this is...
I mean, you certainly see that strongly. I think one of the big problems with that whole resistance mechanistic approach that people have taken, particularly from the clinical samples, is generally well, often it's done in looking at blood, circulating tumor DNA, and the variant allele fractions that people are seeing are always really low compared to the driver mutation. And that worries me. And even when it's much more rarely people actually looking in tissue samples, but it's the same story there. That, you know, so you're not seeing, you've got a level of KRAS G12C mutation of 100, and then you end up with BRAF mutations at 100. You end up with BRAF mutations at 2, and you end up with KRAS G12D mutations at 1, that sort of thing. So I think the amplification of KRAS, that's been harder to sort of get good numbers on, I think. So it is certainly possible that could be a major part of it, an underappreciated part of it. I also do worry with the resistance mechanisms that, you know, it's the sort of the lamppost effect, that people are looking for specific things, but there could be something else that we just don't know about that's really what's taking care of most of it, and we're just seeing the sort of little things, the minor stuff, because that's what we're looking for. But there could be something else bigger. But I think it is true with the KRAS amplification. I think it's true. It's harder to quantify that, and that may have led to it being underappreciated.
I mean, based on this hypothesis, which could be wrong, I'm surprised that with all the KRAS inhibitors that have been developed and you listed, I haven't seen any PROTAC. There's a new one. There's one now that I heard that, although the data, the clinical data is a little bit confusing, but there's not, I mean, that's the only one I haven't heard. Maybe you know more.
Yeah, I mean, so, I mean, well, I guess if it's really a problem of RAS amplification, then I guess any RAS inhibitor, if it's good enough and you can give enough of it so it's not toxic, you might be able to tackle this problem. With the PROTACs in particular, yes. I mean, I guess at ESMO there was the first clinical data from the Astellas PROTAC for KRAS G12D, which didn't look that impressive, but on the other hand, it's, you know, it was first in humans, so it may that may be a bit unfair to expect too much of it. And then we just had published in Science this week the Boehringer Ingelheim PROTAC. I mean, not clinical, just the development of it. So I mean, there are clearly quite a lot of PROTACs around for KRAS, but only that one in the clinic. I'm not sure if they're going to fix the problem. Yeah, I think the only way is to really get good ones in the clinic and see how they do. I can see the appeal of them, but I think it's a bit different from, I mean, RAF, I would much rather see a RAF PROTAC, because I think, RAF, you've got biology where there's clearly stuff that's going on that may not be targeted by the kinase inhibitors. So there you've got a real biological difference between what a PROTAC would do and a kinase inhibitor. But in the case of KRAS, I think it's probably all degrees of the same thing, would be my feeling.
So, Julian, just one question is, in those experiments with the SHP2 inhibitor, with and without, were you able to quantify the clonotypes? So, are those different? The same tumor, you're getting expanded clones when you're inhibiting SHP2 as compared to only when you're inhibiting KRAS? That would be kind of quantifying that you're really getting effector T cells that really hitting the tumor.
Yes. Yeah, we do see that. I mean, we can see the T cell receptor. I mean, we've done TCR sequencing, and we can see the clone types expanding up. It's a little different, the picture you get with the RAS inhibitors and the SHP2 inhibitors. The RAS inhibitors actually tend to get a smaller number of clones brought up. The SHP2 inhibitors seem to give you more clones, but there is this expansion going on in both cases. But yeah, we haven't really sorted out that in full as yet.
Another thing is, we typically combine in the clinic chemo plus immunotherapy in KRAS tumors. Yeah. So the real question in the clinic for us is, are it going to be better to combine a KRAS inhibitor with a PD-1 inhibitor as compared to chemo plus a PD-1 inhibitor? Because in fact, we have known, and we have a lot of preclinical and clinical data showing that you're able to prime and to maintain, at least in a subset of tumors, the immune response by using chemo. So let's say, have you got a control of chemo as compared to a KRAS inhibitor in some of those models?
Yes, I mean, we've done a little bit with chemo, but not extensively. I think it's something that we're certainly very interested in. I mean, we've used some chemo approaches to try to prime an immune response, deplete particular components of the immune system. And I think it's definitely an area that we probably would want to sort of factor into these combinations at some point. I mean, I think I'd be very interested in sort of sequential use of these and how you can sort of get the most out of these sequential approaches. And there, I think at the moment we don't really know what order you would want to do them in. I think if you can start to select up T cell clones that are specifically recognizing antigens on the tumor cells, that would be a very positive thing, and those should be able to sort of survive for a reasonable length of time, where you might be able to encourage them in combination with a number of different drugs. But at the moment, I'm not quite sure which order you would do that in. I think the RAS inhibitors give a very good opportunity to get a sort of a big sort of bang of tumor cell death that is good for priming an immune response. So I think that would be a part of it. But then if that would work better before or after chemotherapy, I'm not sure. But they are things that are addressable in these models.
Thank you. So just last question. So in the model of the immune-excluded RAS tumors, we're using the combos of KRAS, SHP2, plus PD-1 inhibitors. You were getting infiltration into the tumor, nice responses, but the Tregs were going up. Then, when you're adding CTLA-4 inhibitors, you're able to maintain responses longer. How were the Tregs when you're doing the CTLA-4 inhibitors? I suppose they're going down, or?
CTLA-4 is depleting the Tregs in the tumors, but not as well as the CCR8 antibodies. So it's a sort of it's halfway there. But obviously, CTLA-4 is doing other things as well. But it's not as good as the CCR8 to deplete Tregs.
Have you tried like CD25 inhibitors or so?
No, we haven't done CD25 inhibitors. No, my colleague at UCL, Sergio Casado, is doing a lot of that work, but we've not used them ourselves.
So, let's say 1 decade ago, it was very difficult to understand RAS biology, because of the 10 different effector pathways, 12, 15 effector pathways. Now, in your review, you have like 15 new pathways influencing the microenvironment, right? Which is, I would say, frightening. And I can imagine a scenario in a patient where, for instance, if you treat the patient, patient with KRAS G12C mutation, you treat the patient with inhibitors, and now, as you said, there are several mechanisms of resistance, right? So if the patient has an additional mutation in RAS that rescues the whole potency of the oncogene, you might rescue all the downstream pathways and the influence in the microenvironment, right? But in many cases, you have mutations in MAP kinase, JAP1, etc., right? Is there any way or any map of biomarkers to understand, for instance, in that specific patient, whether the tumor microenvironment has been recovered, is still responsive to some immunotherapies? I see that very complicated, right? I get to see this, but so how do you see that in the patient, not in a mouse model or in a cell line?
I think you could certainly, the difficulty is that your starting point for that would be to get tissue samples of resistant tumors, which is already a big ask. And then, if you had that, I'm sure and if you had enough of them, I'm sure you could actually work out some of these connections as to what would be, biomarkers for something where there would be some potential for combination responses. But I think the difficulty of just getting that sort of cohort of samples together would be huge. And as you say, it is just very complicated, and it's not that easy to figure out what's really going to make a difference. Obviously, an appeal with the mouse models is that you can do all these sort of studies and you can get all this information very easily. But the big problem is they're mice and they're not people. So you really, ultimately, the tests end up being clinical trials that come out of this.
So maybe I'm just going to do the last question. Sure. So what is the mutational burden of those mice models you're using? And the question is, what would happen if you, on top of that genetic component, you really try to make those tumors a bit more closer to what happened? Let's say, put a lot of mutations there, and
Yeah, we spent a lot of time trying to do these things. One thing was using carcinogens in the mice, and Mariana's done this as well. You can get with urethane, you can induce lung tumors and get a reasonable mutational burden in them. Although actually we did find that the mutation burden in those tumors was not really as close to as much as you'd see in a typical smoker's lung cancer, but it was a significant tumor burden. So I think you can you can sort of build up mutations in some of these model systems. You can do it in vitro and then do transplants. The approach we took with the engineering in a DNA changing molecule, which was what we did with this cytidine deaminase APOBEC 3B. So APOBEC was appealing because lung cancers have a mutational profile that suggests there's been this cytidine deaminase activity in them. And so we thought by overexpressing APOBEC, we could cause that, and that would increase. And we did increase the tumor mutation burden, but something that just the way we designed it was, looking back on it, was not the way we should have done it. But what we were getting was a huge number of subclonal mutations because you were putting in, the enzyme was introducing mutations as it went along, and then the tumor cells would divide, and then it would introduce more mutations. So what we got is the tumors where if you did bulk sequencing, you couldn't really see that many mutations. But if you took cells and took single cell clones, then you could see they had lots of mutations. But that's a hugely heterogeneous tumor mutation burden you're generating there, which are notoriously not good for immune checkpoint therapies or any other immunotherapy.
Thank you very much for your talk, and I think it's time for coffee. And I think we are having like 30 minutes, so we can come back at 12:15. Okay, thank you. Lunch, and I'm glad to present to you today Dr. Malumbres. Marcos is the guru of cell cycling, and he's going to tell us how to take advantage of cell cycle disturbance in lung cancer.
Okay. So thanks, Luis, and the Fundación Ramón Areces for this great opportunity to be here discussing some ideas. So we are part of the iLUNG consortium, and we are very interested in applying our knowledge on the basic regulation of the cell cycle to discuss some possibilities for cancer therapy in this disease. So let's say the main goal of the laboratory is to try to understand which are the critical proteins that control the entry into the cell cycle, and therefore they are responsible for getting most of our cells, which are quiescent in our body, into the cell cycle and start proliferating, which is a hallmark of cancer. So the critical idea is that there will be some signalings. Sorry, I need to. Okay. Oh, I see. So there are major mitogenic signals in the cell that somehow trigger the cell cycle machinery just to get into cell cycle proliferation, right? So that happens obviously in every normal cell. This is critical during embryonic development, etc. But at one point there is something different that is downstream of oncogenic signals that in principle might be different from the normal mitogenic signals, right? And obviously the question of what is different between one and the other is a major question, and I don't have the answer. I don't think we have the answer at all. I cannot think of any single therapy in the clinic that is able to discriminate one versus the other. But the point is, okay, so which are the main regulators of the progression into the cell cycle? So there are many proteins with enzymatic activities, such as kinases, that are involved in cell cycle regulation, but there is a specific family named called cyclin-dependent kinase, whose kinase activity depends on binding and activation by a cyclin subunit. So this is a complicated family. There are at least 20 family members, and in the last years, obviously, there have been a lot of efforts to investigate, which one of these proteins is downstream of a mitogenic or an oncogenic signal. So we can somehow predict what is the relevance of these kinases using a tool which is available for everyone online, which is what is known as the DepMap. So this is a dependency map based on CRISPR studies in which people have analyzed what is the effect of knocking out each of these proteins or any gene in the genome in more than 1,000 cell lines, right? So now you can take the different cell lines that belong to breast cancer, prostate cancer, bone cancer, just get the average, and you get this profile. So what you see here as the dependency index, the more negative the number is, the more profound the effect of eliminating the gene is in the cell proliferation, right? So that means, for instance, that CDK1, which is the first one over here, this is a -2, again, this is a relative number, but means that every tumor cell line is very sensitive of eliminating CDK1, right? So most of them stop proliferation or die when you eliminate CDK1. Again, this is an example that you can see, the example of what is the consequence of eliminating CDK2, any of the others. I have to say, again, every dot is the average in a tumor type, so meaning that, for instance, lung cancer is one of the dots over here, and is represented for about 65 cell lines, right? So this is a lot of data here. It is also very important to consider that it's a dot which are normal non-tumoral cell lines. So that means that, for instance, CDK1 is as toxic in normal cell lines as it is in tumor cell lines. And I'm afraid that happened to almost every family member. Right? But again, if you analyze this, for instance, specifically with CDK4, you see that the tumors which are more sensitive to CDK4 are, for instance, breast, prostate, and bone. And it is not surprising that actually CDK4 inhibitors, together with CDK6, because they are very similar, as you see here, these inhibitors of these family members having approved for treating breast cancer tumors, specifically hormone-positive breast cancer tumors. So it's somehow you could predict that from the map information in cell lines, and this is why I'm using that as a starting point for some of the discussions. So these are the CDK4 inhibitors which are already approved only for breast cancer, and as you see in this cartoon, I took this data last year. There were more than 500 clinical trials with the name of CDK on them, because the number of CDK inhibitors for the different family members, but mostly because of the number of different combinations, trying to understand which is the relevance of eliminating CDK4 together with MAP kinase, together with RAS, together with immunotherapy, with or without rationale, because at the end, these clinical trials are really fast compared with biology. But again, there is a lot of interest in this family, and you see in blue the ones which are approved, either in the States, Europe, or Asia, and the ones that are now in clinical trials, and obviously the list is growing and growing. Now, we took advantage of this information in that map, and this is the work of Agustin, a PhD student in the lab, to try to do a map of the relevance of each of the genes in the genome, but with a focus on the CDK family, right? Why this interest? One might think that if the cell cycle needs to be activated by a member of the CDK family, if you have a tumor which is resistant to CDK4 inhibitors, it might be sensitive to CDK12, for instance, right? So the critical question is how tumor cells, or how the cell cycle machinery in tumor cells is rewired in response to current therapies. For instance, this is a way that I really like to represent this data. Again, the dependency index represents how strong is the elimination of the gene. The more negative, the stronger the phenotype. For instance, again, you see CDK1 is to the left, is the more dramatic effect in cells. And in the y-axis, what you see is selectivity. Selectivity means that when the selectivity is very low, this effect is spread through all tumor types. When the selectivity is high, it's specific for some tumor types. Right? Again, CDK4 is a general toxic, or CDK4 elimination is very general, is part of the non-specific, so high dependency in all tumor types. So I would call these are essential genes, right? I'm not saying that an essential gene is not a good one, a good therapeutic target. There are many examples, actually most of them. But there are some genes where the index is very high and the selectivity is also high. So you might select some tumors where it works, some tumors where that does not produce any toxicity, and you might expect that maybe in the normal tissues, the toxicity might be very low, right? So from this analysis, you might predict that maybe the ones which are in the top panel over here, especially the ones to the left, they are very good targets, and maybe the toxicities are less than in the ones over here. And again, I think this prediction is somehow correct, because again, cyclin D1, which is the guy over here, is the main partner of CDK4, and is the main target of the CDK4 inhibitors, because the two of them form part of the heterodynamic complex of CDK4 cyclin D1 kinase activity. So again, you have a combination. So I'm representing all the genes in the genome as blue dots, but only the CDK and cyclin family as blue, with the names, because I'm interested in checking those, right? One application of these possibilities is, for instance, to try to answer the question, again, how this is rewired in a specific scenario before and after progression to a therapy. What I'm showing here is the plot in breast cancers. So these are all the breast cancers, well, all of them, these are 19 different cell lines which are sensitive to CDK4/6 inhibitors, specifically the ones that are sensitive. And again, as you see here now, it is amazing, how actually cyclin D1 and CDK4 are top to the left, even more than CDK1. So, these tumors really depend on cyclin D1 CDK4 complexes, right? So now the question is, okay, but now if you get these cell lines resistant, how the machinery is rewired. And so this is not the plot. Again, what you see here, CDK1 is still there. It is very difficult to move CDK1 from this position. And now you see a few changes. For instance, cyclin D1 and CDK4, obviously, this is the control. Now they are to the right. These cells are resistant to eliminating these genes. And now, among the few changes that we can see, for instance, CDK2, which is over here, it's like -1, now it's -1.5, well, fine. there are some increased dependency on CDK2. I have to say that there are a number of companies which are now crazy about using CDK2 inhibitors, although the evidence in vitro is, I would say, low. But again, I think this is a possible representation to identify some candidates. For instance, we also see increased dependency on CDK9, which is a transcriptional CDKase, and I will mention this later in maybe more difficult tumors. Now, if we compare what is different in these cell lines, again, these are breast cancer cell lines, which are resistant to CDK4 inhibitors or sensitive to CDK4 inhibitors, we can now ask, the ones which are resistant to the inhibitors, now they are more sensitive to what? So which are the new therapeutic opportunities that appear in a cell when become resistant to CDK4? And we have a few candidates, which are shown here, obviously including cyclin E1 and CDK2, also CDK9, and some others which we are trying to validate in the lab with certain accumulation of genes involved in the Wnt and MYC pathway. Again, this is breast cancer, but from very old, very classic and pioneer research, and including by Mariano Barbasi's lab, we know that there are some other tumors that might be sensitive to these therapies, including, for instance, non-small cell lung carcinoma and others. So we did a similar assay, taking now a number of lung cancer cell lines. In this case, we have 18 lung cancer cell lines which are sensitive to CDK4/6, and 7 which are resistant to CDK4/6. Obviously, the term sensitive or resistant, is a gray scale. We need to classify, with a threshold, but we have quantitative data, right? So this might be more complicated. But now, if you analyze what is different in the sensitive ones, again, that's good. The sensitive ones depend a lot on cyclin D1 and CDK4. Cyclin A2, which is major cyclin activating CDK1 is here. Everything, fits, right? Now, what is actually depressing is that in lung cancer, the ones which are resistant to CDK4, is very difficult to see any change in the cell cycle that accounts for this, right? Obviously, there are many explanations. I would discuss a few of them, but I would say, there is not obvious candidate to be used in lung cancer in this case. Now, obviously, if now we compare the spread, so again, the dependencies, same question. These lung cancer cell lines which are resistant to CDK4, now they are sensitive to what? Actually, it's a bit surprising. It's not fully surprising, but we see a lot of neuroendocrine markers here. Obviously, you might think that the neuroendocrine markers might be a reason for the resistance. You might also think that typically neuroendocrine tumors, they are p53 null, RB null, right? But it's not that they are null. They also are dependent on the neuroendocrine transcription factors. Again, this is something we are now trying to understand and validate, but actually we were very surprised, and actually I'm showing this slide, because among these candidates, we found this molecule here, which is MASTEL. So MASTEL is a microtubule-associated serine-threonine kinase. It has been published by Tim Hunt to be the inhibitor of PP2A, which is the main phosphatase that counteracts CDK activity. So let's say in the cell cycle in the cell, CDKs promote proliferation, and PP2A shut down the activity of the CDKs, right? So this is the balance in the cells, CDKs, PP2A. And MASTEL is helping the CDKs, just inhibiting the phosphatase. We were interested not only because of this data, because we have been working in the last 15 years with this molecule. So we discovered actually MASTEL as a critical regulator of mitotic acid. We generated a knockout. We generated data supporting its role as a therapy. And we identified mutations in human thrombocytopenia, et cetera, et cetera. But we almost stopped this project because we never found a clinical scenario where MASTEL provide any advantage, right? So now we have a candidate that probably is small cell lung cancer, and we are trying to validate that in the laboratory. Now, as I said, there is a lot of interest in the family, right? Remember, there were inhibitors already approved for CDK4/6, a lot of inhibitors in clinical trials for these family members, for these others over here. So these are the transcriptional CDKs because they mostly regulate RNA polymerase II. But there is a specific family that brought my attention many years ago. Actually, when preparing this figure, to me was very critical. I always use this plot because if you check the sequences and you get this tree, you always get this family over here, which are very close among them, and very close to CDK4 and CDK2 and CDK1. And unfortunately, there are almost no papers about this family. One of the reasons, actually, you can get a hint why is that, comes from that map. What are the consequences of eliminating CDK14, 15, 16, 18 in cell lines? Again, in more than 1,000 cell lines. So the plot is here, 0, right? You get no effect if you remove CDK14 or CDK15 or CDK6 or et cetera, et cetera. Obviously, I think that probably might eliminate any interest in this family, but one thing I learned with Mariano, studying this family, is that there is a lot of compensation, right? They overlap a lot, and they are very similar. For instance, CDK4/6 inhibitors, they are called CDK4/6 inhibitors because they inhibit the two of them. Although in breast cancer, the target is CDK4, but you cannot separate these activities, right? They are so similar. So that means that if you get a CDK16, 18 inhibitor, it will inhibit the three of them. It's going to be very difficult for the chemists to separate these activities, right? The other thing we learned when we generated CDK2 knockout, CDK4 knockout, CDK6 knockout with Mariano, is that the CDK4 knockout is alive and quite happy, the CDK6 is quite happy, the double for 6 embryonic lethal, right? So we thought, okay, maybe why don't we knock out not a single family member, but combinations? Obviously, these data are not available in the map because in the map you only have single knockouts, okay? So what we did, so actually Diego was very brave to start his PhD project with this because it was a very crazy idea and very laborious in the laboratory to start generating individual knockouts, double knockouts, or triple knockouts. So what he did is to start doing CRISPR. So this is single guide RNA for CRISPR guide elimination of CDK14, 15, 16, 17, 18, double 14, 15, blah, 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 all the other, and even triple, right? And now you see this data. I mean, if you eliminate two of them, for instance, 14 and 16, or you eliminate 16 and 18, et cetera, now you do see a strong anti-proliferative effect. This example is in liver cancer, but I will show you more data now. Again, don't pay attention to the statistics, but I think the images are representative enough for the phenotype. And probably this is even more dramatic and more obvious. So this is a colony formation assay, eliminating only CDK16, no differences, 17, 18, some differences. Really, this is an assay that lasts for almost 2 weeks in vitro, so you accumulate more effect. Still is very mild. And this is the effect after eliminating 16 and 18, right? So actually, we got single-cell clones knockout after this CRISPR exercise. We sequenced them, so we know the genetic alteration. And actually, in the double knockout, it was very hard to maintain these cells alive. I mean, there is no way they can actually grow. Actually, we tried to generate the triple 16, 18, 17. These are very similar family members, but it was impossible to put the CRISPR reagents for CDK17 in these cells over here. I mean, they are not growing at all. Now, I'm not presenting most of my molecular data. Again, there is very little information about this subfamily, but we identified using proteomics, etc., that actually these proteins, I'm talking now about CDK16 and 18 mostly, which are the critical targets in here. These proteins bind and phosphorylate several members of the nuclear pore complex. We identified, actually, we validated, and we have antibodies for the phosphosites. So we know that these molecules phosphorylate these proteins. And what is the consequence of inhibiting these proteins? So after elimination of CDK16, 18, or when using a small molecule that actually inhibits this subfamily. So this molecule, we call it FMF. FMF comes from the name of the postdoc in Nathaniel Gray laboratory at Dana-Farber at that time, who generated this molecule. This molecule inhibits this family, but it's very dirty. It also inhibits CDK2, CDK9, and others, right? But we validated this with CRISPR knockouts and inhibitor, and actually, when you eliminate this family or you inhibit this family, you produce a disruption in the normal regulation or the normal structure of the nuclear envelope. I mean, we cannot say that this eliminated, but it's certainly disrupted, as you see, for instance, in here. This is a plot for one of the markers, or in here. So I would say the nuclear envelope is kind of leaky or partially broken, and actually cells accumulate some defects in the nuclear envelope. Now, what are the consequences in terms of therapy? Now we know that when we inhibit this family, again, either using the inhibitor or using CRISPR knockouts, so using the clean genetic knockouts, I think this slide is complicated, but it's very simple to exemplify the phenotype. So these are normal cells, and we are representing here a marker for homologous recombination, RAD51, so for DNA repair, and a marker for DNA damage, right? So in a normal cell, non-treated, obviously this is a happy nucleus with no signal for damage or for repair, sorry, for damage in red or for repair in green over here. If we use olaparib, which is a PARP inhibitor, this is a molecule that obviously induces DNA damage in the cell that cannot be repaired because it's inhibiting PARP. In that case, actually, we see accumulation of DNA damage, and immediately that correlates with concentration of the proteins responsible for homologous recombination repair in the cell, for instance, RAD51, right? So in a normal cell, when you induce damage, immediately you have DNA repair machinery in the nucleus, concentrated to repair. Now, when we inhibit this subfamily or the knockout, actually, by itself, that induces a bit of damage, right? So we have damage only by knocking out the genes of using FMF, and FMF is dirtier. And now there is no accumulation of RAD51. Even what is more dramatic, if we use olaparib, and now cells depend on homologous recombination, even in that case, which is very specific, actually the accumulation of RAD51 is defective in the cells, in the absence of this protein. So again, we have evidence that actually if we put a dye into the nucleus and we knock out these genes, actually the nuclear envelope is leaky and the dye gets out of the nucleus. So it's not only RAD51, it's a general defect in the nuclear envelope. Again, this is a mechanism, but now the critical experiment is, okay, can you eliminate the tumor with these family members? In the past, and again with Mariano, actually, this is the cartoon of the cell cycle family of CDKs, including CDK1, CDK2. We generated these knockouts with Mariano in the past. Every laboratory mouse animal is a natural knockout for CDK3, so you don't need to knock out CDK3 in these animals, and also CDK4 and CDK3, right? So CDK5, what also generated by different groups, is mostly a neuronal kinase. So we decided to generate knockouts for the missing family members. So in the past few years, we have generated five different knockouts, and now we have crossed, and we have a lot of alleles combining to try to eliminate one side the CDK16-18 family, on the other side the 14-15 family. And we select this experiment. So this is a various experiment. This is typically very well set up in the laboratory of Dado, in which you can inject oncogenes, and we use in this case MYC and beta-catenin in the tail vein, and these oncogenes go to the liver and they induce liver tumors. Actually, we detect that in that case, using just wild-type animals, actually CDK16 was upregulated in these tumors, and that also happened in human tumors, and we have this evidence, right? So the experiment that we did is, number one, to inject the oncogenes, which is the hydrodynamic injection over here in the tail vein, now to analyze the mice by imaging, because together with the oncogenes, we put luciferase in the cocktail, so we can see how tumors evolve, and now either use the inhibitor or the genetic knockout. So this is tamoxifen for the genetic elimination of the genes, right? Now, the inhibitor is very dirty. For ethical reasons, we can only use the inhibitor for 1 day, 2 days. Again, it's very dirty. It's not only specific inhibitors, but when using that, actually, very sorry. Let me start with the knockout. So this is the number and size of tumors in the liver in the control, and this is the size and the number of tumors in the animals after eliminating of both 16 and 18. I have to tell you that every animal was treated after the tumor was visible by imaging, right? So this is not preventive. This is after the tumor was already identified. And actually, this was accompanied by a lot of DNA damage. Again, this is the same marker you saw in the previous slide for DNA damage. In this case, it's caspase. MYC just differentiate the tumor versus the adjusted liver, normal tissue, which is normal and is not suffering from the therapy. Actually, only the tumor is suffering from this therapy. Now, the summary, I'm finishing soon. The summary is, we think we have a new family, with very little information. Maybe the trick is, in this case, you need to inhibit several family members. But again, no problem with that. It's going to be very difficult for chemical laboratories to design inhibitors for just one or the other. So most of the inhibitors will inhibit the three of them. Right? So when you have tumors with a normal DNA repair, with a normal DNA repair, I mean, you might have DNA damage in the tumors for different reasons, but eventually you have repair, right? So in the case that you inhibit or eliminate CDK16/18, we have this problem in the nuclear envelope and defective DNA repair because of the lack of proper nuclear pore complexes, and then we increase the lethal levels of DNA damage until most of the cells die because of this problem. Actually, we know, and this is important now for my final slide, we know that this phenotype increases in tumors with high Wnt, high MYC. To me, this was the best expectation in this project, because I always use an experiment by Peter Sithiski where he analyzed breast cancer tumors by eliminating cyclin D1 in the tumors, and the ones which are RAS positive or hormone positive, they respond very well, but the Wnt1 or the MYC1, which are very aggressive oncogenes, typically of triple-negative breast cancers, CDK4 has nothing to do with Wnt and MYC. In this case, I think we have evidences that actually, in the presence of Wnt, of high Wnt or high MYC, actually the elimination of these inhibitors works even better. And I will skip this one. And one exercise we did is to try the elimination, for instance, of 16/18 in a panel of 25 different cell lines representing 9 different tumors. And for instance, you will see here that, again, liver cancer, well, I need to explain that CDK1 is a technical threshold of the experiment, right? So if you have a cell line in which the gray one, CDK1, is close to the red one, 16/18, that means that the elimination of 16/18 is as strong as CDK1, which is the stronger control I can get, right? Well, you can see here that actually this strategy works actually very well in liver cancer and also in lung cancer, as you might see in these examples in here, right? So one exercise that actually Fatima did, and she's in the audience, I will be presenting this afternoon, is actually testing those inhibitors maybe in the most aggressive tumor we can think of, which is a small cell lung cancer, which is P53 null, RB null. And again, I mean, we have evidences that, let me just go very quickly through this slide, for instance, in here. So this is proliferation in the control. This is proliferation when we eliminate CDK1. Again, this is genetic using CRISPR. And this is the proliferation when we eliminate a combination of 16, 17, 18. Now, remember, these 3 family members are very similar, and simply the question of which one is the target, either 16, 17, or 18, just depends on the cell type and the expression levels. But again, probably any chemical will inhibit the 3 of them. So, I mean, this is very preliminary, but I think we are very interested in, going ahead with this idea in a collaboration within the iLUNG consortium and with Luis' team in other consortiums. So the summary is the following. So, there are a lot of evidences in vitro, in preclinical models, that CDK4/6 work in lung adenocarcinoma. I think the evidence in vitro is huge, and they don't work in clinical trials. Right? So this is a huge question. I think there are evidences, and we have some evidences that actually CDK6 is able to bypass the effect of the current inhibitors. I'm referring to palbociclib, ribociclib, abemaciclib, the 3 of them. We are working on that. We have some ideas, and we have a couple of candidates. And we also thought that, eventually these patients that will go to chemotherapy, and we might have some combination strategies with chemotherapy, and actually Fatima Guerra will be talking about that later today. And finally, I think we have some ideas about using the other members of the family, mostly transcriptional CDKs. I didn't mention those today, and also the CDK family in this very aggressive disease, and I hope we will be able to provide more data in future meetings. So with that, I will stop. So these are the collaborators. In brown, you have the people in the lab that I already mentioned, including Begoña Hurtado, which is also with us today, involved in this project, and the collaboration from the iLUNG community. So thanks a lot, and happy to take questions.
Any questions for Marcos?
Marcos, I was surprised by your last slide. Why CDK6 does not respond to CDK4/6 inhibitors? I mean, what is needed to make a real CDK6, to inhibit CDK6?
We are working on that. I'm afraid that the answer to that question is worth a lot of money. We are working on that. There is evidence, there is published evidence that actually CDK6 might be protected by another molecule, so the inhibitor does not get into the pocket, right? And this is published. There are evidences. It's not clear what is the cause of that. So we have a project in the lab working on that. We have some candidates, and we are performing screening. We have a couple of candidates, and we are working on that. So I cannot tell you that we have the answer, but I do believe it's a very important answer for many tumors. So if we got CDK6 back to be responsive to these treatments, I think a lot of therapies will change a lot. I tell you that, for instance, in breast cancer, the immediate resistance to CDK4/6 inhibitors is CDK6.
Hi, Marcos. I had a question about this data on small cell lung cancer. Do you have any idea of how much the effect of knocking out these CDKs might depend on the deficiency or proficiency of RB1?
RB1. Yeah, actually, I have one slide, but I don't need to present it. So we actually did some analysis to check whether we could answer that question, not only with RB1, but also with p53, with the neuroendocrine markers, et cetera, with high MYC, et cetera. We didn't find good data from the cell lines. So we would love to answer that question using data from patients. From the cell lines, I cannot tell you that this is, is dependent on the absence of RB1 or is dependent of the presence of MYC1. Might be a combination, but we didn't get a good single hit to answer that question.
All of them seem similarly sensitive to the knockout?
No, there is variability, but we don't find the proper biomarker for the variability.
Marcos, the fact that some tumors with CDK6 amplification have a strong expression of PD-L1, do you think it opens treatment opportunities? I know it's very anecdotal evidence, but at least in squamous cell carcinomas, not only of the lung, but in other locations as well, that's a very, I think, established link. What your thoughts on this possibility?
It's very obvious. We knew with Mariano, we published a paper in 2003 reporting that CDK6, most of the biological role is in the immune system, in hematopoietic cells. Most of the toxicities that are now in patients were in our mouse model 21 years ago, right? And this is because of the relevance of CDK6 in the hematopoietic system, right? Now, there are a lot of evidences on how, actually there are several papers on how CDK6 And some of them are very confusing, also involving CDK4. I tell you, there are like 4 papers in Nature, and the 4 of them report a different mechanisms, right? So, I mean, we are pretty sure there is something. I don't think there is a consensus what is the role, but I do, let's say, I could confirm or agree that the data in vivo of inhibiting CDK4/6 and the consequences in making a tumor hot are very solid. So I'm pretty sure there is something, but we don't understand well the mechanisms. I think there is a lot of controversy there. Yeah.
Marcos, looking at, I mean, when you make your cells, let's say, the lung cancer cells resistant to the CDK4/6 inhibitors, actually the transcriptomic profile look like, at least some of the genes you show, very much as a tumor that is evolving to non-neuroendocrine subtype of small cell lung cancer. That being a non-small cell lung cancer cell line, so somehow you're inducing cell plasticity.
Only one comment. So these data are not really transcriptional profiles. So this is not expression of genes, is the dependency on the gene when you knock it out. Okay, so that means I don't know whether NeuroD1 is more expressed in there, but the cell is more dependent when you eliminate NeuroD1. So these are CRISPR data, not expression data.
Actually, the question was... So that was the question. So what is the transcriptomic profile of those cells? So are those looking like, and the second thing is, if you do that experiment on a in vivo model, those typical genes are accommodating a microenvironment that actually is very inflamed as compared to a very immunosuppressant microenvironment to start with.
Yeah, so I think the second part is impossible to answer with the cell lines. For the first part, actually, we have discussed with Álvaro Quintanal some ideas because he has been working with this transversion from one phenotype to other phenotype in cell lines, and we have some ideas with him. Yeah.
Any other question? Okay, so thank you very much. So now Professor Mustiano is going to tell us what to do with the KRAS patients, with the KRAS tumors, let's say.
Okay, so thank you, Luis. And I would like to give a talk on, basically, my subtitle is Restoring Interferon Signaling to Enhance the Efficacy of Immunotherapy. And what was the outcome? So after Julian's presentation, I don't need to introduce any more lung cancer, just to point out that there are different types of lung cancer, as we heard in the previous talks. So 15%, they are the small cell lung carcinomas, and the other 85%, they are the non-small cell lung carcinomas, from which 40% are the adenocarcinomas, in which we are focusing in the lab, basically. And from this 40%, as Julian already mentioned, we have like between 10 or 30% that harbor KRAS mutations. So we heard already as well that the most common one is the G12C mutation, where there is already a drug, so these KRAS G12C inhibitors. And the second most frequent mutation in lung adenocarcinomas is the G12V mutation. Just pointing out to this is that this is the model that we are using, the mouse model that we are using in the lab. It's carrying this G12V mutation. And in terms of 5 years survival, so it's pretty clear, most of the patients, they are diagnosed with distant metastases at stage 4, and the 5-year survival is less than 10%. This we already heard today, that there is lots of drugs. The drugs that Julian mentioned, the KRAS G12C inhibitor, the KRAS1 inhibitors, I just mentioned two, directly KRAS inhibitors, but this slide is from 10 years ago, so you can see here that there is lots of pharmacological compounds to target the downstream effector pathways of KRAS signaling. Unfortunately, for KRAS mutant tumors, none of these inhibitors have shown very significant benefits in patients, and basically mostly due to toxicities or resistances, as in the case of the latest developed KRAS inhibitors. So seeing this in the lab, just to mention, we have a nice target, that's CRAF, where we show that elimination of CRAF really induces lung cancer tumor regressions. Unfortunately, we do not have yet a drug because we do not need a kinase inhibitor. We would need a PROTAC, so we are currently working on that. And since we do not have a drug, we were thinking, let's see what we do to find new targets to treat KRAS mutant lung cancers. So for that, we performed single-cell RNA sequencing, taking advantage of so crossing our mice, the KRAS, lox-stop-lox, G12V model that we are using in the lab with the ROSA26, lox-stop-lox, EYFP mice, in order to have the cells labeled with the yellow, enhanced yellow fluorescent protein to be able to isolate the cell type of interest. As controls, we were using the KRAS wild type littermate, harboring as well this YFP protein. So at 8 weeks of age, we infect these mice with adenoviral particles encoding for the Cre recombinase, and after 4 weeks, post KRAS G12V expression, we sort for the 82 cells, taking into account that the 82 cells are the cell type, the lung cell type that would, from which these tumors, the lung tumors arise. So by FACS sorting for EYFP positive cells, obviously excluding the endothelial cells, the lymphocytes, and marking for MHC class II positive, because these are cells that they really express the MHC class II complex. We sorted the cells from these two mouse models, the controls, KRAS positive, KRAS G12V, and performed single-cell RNA sequencing with the C1 Fluidigm platform. That was the prior one to the 10x platform that most of the people use now. So the first surprise that we got when we analyzed the data is that we could perfectly cluster the cells in just two clusters. On one hand, the KRAS wild-type cells, and on the other hand, the KRAS G12V expressing cells. Here I just marked this gene, very little is known about it, but only with the expression, with downregulation of the expression of this gene, we were able to cluster these, to get these two clusters. I was waiting to find more clusters, because not all the infected cells or all the cells that are KRAS mutants would give rise to a tumor, but we only clustered the cells in two clusters. After further analysis, what we managed to get from the single-cell RNA sequencing analysis, so 3 distinct aspects of transcriptional heterogeneity. In the aspect 1, we had gene sets with increased expression in KRAS mutant 82 cells, all, how you can see here, related with genes related to translation, etc. So everything pointing out that these cells, they are more proliferative, so all the pathways upregulated that had to do with proliferation. And aspect 2 and aspect 3, they were gene sets with decreased expression in KRAS mutant 82 cells, from which the most prominent was the aspect 3 with the type 1 interferon signaling pathway downregulated, completely downregulated. So the second surprise was when we went to the gene of the list and we said, let's see what differentially expressed genes we have upregulated. And there the surprise was that we saw some some long non-coding RNAs. The only known gene was cyclin D1, and few other more that are basically untargetable. So we decided to continue investigating the genes that were, to investigate the differentially expressed genes in this aspect 3. So as you can see here, basically most of the differentially expressed genes significantly found to be significant. They played a major role in interferon pathway, like STAT3, STAT1, for example, IRF7, and lots of molecules involved in antigen presentation. They were completely downregulated, as pointed out here in this plot. So next we wanted to, this is not very novel, it was already described that, that KRAS induces immune evasion, but we wanted to see if this, we went further to see if this occurs in different genetic contexts, and with this I mean in different tumor models in which we generate more aggressive tumors by eliminating p53 or LKB1, and if this occurs in advanced stages in our mice, so to be sure that this downregulation of the gene is is present still at the moment of the treatment. And finally, we wanted to confirm if the human data in the TCGA database confirms these results. So what we did, we harvested at human endpoint tumors from these 3 different mouse models, so only the KRAS G12V expressing lung cells, the tumors that are KRAS G12V and p53 knockout, and from the tumors that are KRAS G12V and LKB1 knockout. And what we could see here is that all the genes that we saw significantly downregulated in the single-cell RNA sequencing were confirmed to be still downregulated in these full-blown tumors. And very similarly between these 2 models, independently if the mutation, if we lacked p53 or we lacked LKB1. Further on, we went to the TCGA database. We took our list of genes that we found involved in the interferon pathway signaling, antigen presentation, and we looked for them in the expression data in the TCGA database. And what we could see here, it's the same, that only KRAS mutant tumors, KRAS P53 mutant tumors, KRAS LKB1 mutant tumors, not to exclude that these are human tumors, so they have more mutations, not only the ones that I depict, but we just stratified between these 2 subtypes. Basically, all of them, they show the same tendency of being downregulated as in the mouse model. I have to point out that as control, we made the same exercise for EGFR mutant tumors as KRAS wild type subtype, and this is true that the interferon signaling is downregulated as well in the EGFR pathway, and only 14 genes are really KRAS specific. So now we wanted to see next, just looking at the type 1 interferon signaling, if there is since we know that there are molecules already in the clinic that they try to restore the signaling, to see if we were really able to restore interferon signaling, but specifically by restoring the gene expression of the genes that we identified. So just to show you, we concentrated at this location where we know that poly-IC, it's in the clinic in clinical trials. It's a agonist of the Toll-like receptor. So like the triggering expression of type 1 interference. We concentrated at this point in activating STING, that it's activated by cytosolic DNA presentation, again enhancing the production of type 1 interferons. And here there are compounds as well in the clinic. And we concentrated on the interferon receptor by where there are already in clinical trials agonists of the interferon receptor that's upstairs of the signaling by STAT1, one of the genes that we saw completely downregulated in our RNA single cells analysis. So by saying that, we took our cells and we challenged them. We took our cell lines, the KRAS expressing, KRAS P59, KRAS LKB tumor cells, and we challenged them with interferon beta as a control, the natural ligand in the interferon receptor, with Rho191, poly-IC, and the STING agonist, DMX-AA. And what you can see here, I just point out to 6 genes, like for example, IRF7. We can see here that interferon beta, it's the agonist, poly-IC, can restore the expression, as well as, for example, the STAT1, that's the final transcription factor that would induce the expression of the MHC class 1 molecules, for example. In addition, we went on to see what happened with the immune checkpoints. We analyzed up to 17, but from which one only like significant increase we could see in PD-L1, HVM, and GAL9. So if you restore hearing of the interferon signaling, it's possible, and specifically restoring the expression of the genes that we saw downregulated in the RNA sequencing. So next we wanted to move further and to see what happens if we restore in the proper lung tumor cell this interferon expression, so the interferon signaling by 2 aims, by direct interferon beta expression, or the expression of a constitutively active STING isoform, and enable to have a model to see if this would synergize with anti-PD-L1 with anti-PD-1 therapy. So we did. We infected our cells with this construct. As you can see here, in both cases, even expressing interferon beta and STING, we can get induce in interferon alpha response. And when we were looking at MHC class 1 family members and the PD-L1, what we could see here that only in the case of expressing interferon we could see these 2 proteins on the membrane of the cells, but STING failed completely to induce these genes. Another interesting issue is that in this case, as you can see here, again, both KP and KL models, they behave the same in the murine model. Next, obvious experiment, we took our infected cells, we implanted them subcutaneously in syngeneic mice. After 10 days, when the tumors reached around 150-200 cubic millimeters, we were starting feeding these mice with doxycycline because our constructs, they are doxycycline inducible. So we can induce interferon beta and STING expression, and after 14 days, after 2 weeks, we performed flow cytometry to see if we saw changes in the microenvironment. And indeed, as it was expected, we saw like enhancement in T-cell infiltration, CD8 positive, CD8, CD4, natural killer cells were enhanced. Another, the dendritic cells, they were enhanced, everything like as expected. So we went on and we took the same model, but at this time, after induction of interferon and STING expression, we started the treatment, as these arrows point out, with anti-PD-1. And what we can see here, that as we already knew, the tumors themselves, they do not respond to anti-PD-1 treatment, so the clear circles. because the tumors I didn't mention, they are lacking completely, they are very cold tumors, both P53 null and LKB null. We hardly see in a normal situation T-cell infiltrations in these tumors. So as expected, they do not respond to PD-1 treatment, but the combination, either expressing interferon in the tumoral cell with the PD-1, the red dots, it's significantly increased. So having said this, this is basically a proof of concept that we pharmacologically it's possible to restore the expression of a set of genes involved in interferon signaling or in antigen presentation. Now it's the time to move on, and we are trying to find novel targets that by CRISPR-Cas9 screenings, the inhibition would mimic the same effect that expression of interferon does, for example, because interferon it's impossible to be given to the patients. Or another approach using the tools that we have generated from these cell lines and these cold mouse models that we have, like doing a high throughput screening of FDA-approved drugs for repurposing issues as a possible extra molecule to a combination therapy just to maintain the expression of the downregulated interferon-regulated genes. So that would be basically the future plans. And with this, I want to acknowledge to the whole experimental oncology group, especially to Mariano, that give me the possibility to be part of his group for the last 11 years already. And to Fernando Fernandez Garcia, the first author of the work that I presented, and to all the core units at the scenario that were helping us, and to the financiation that we got from the iLUNG, ERC, and the Ministry of Science. Thank you.
Question for Monica?
That's very, very nice work, Monica. I'm wondering with the STING expression, the STING agonists, so that was in the tumor cells you were expressing that. So I mean, what do you see the potential for activating STING in it therapeutically, where obviously in this situation you've been able to activate it specifically in the tumor cells, but the problem of other responses to STING activation that might occur. So the problem of activating STING systemically versus in the tumor, what do you think the potential for STING agonist treatments are in the clinic?
The potential of the STING agonists, I'm not very sure, because I still don't understand very well by which mechanism they work, because the STING agonists and the MXA that I was showing failed in the clinic already because it was shown to have an effect in the mouse, but it was incompatible with the human STING, or in the humans. And we do not see expression of the downregulated genes. It doesn't revert. The only thing that it does, it's only attracting chemokines. It's upregulating from the chemokines that we checked, CXCL10 and CCL5. So I think it has a potential, but I don't know what to expect.
Thank you.
Any other question? So you said that interferon, I mean, you can give interferon in the clinic, right?
As far as I know, it's very toxic.
Well, interferon, particularly interferon beta, as compared to chemotherapy, is like Coca-Cola, So. So, I mean, it's not easy, let's say, but it's not really that bad. So, I mean, still workable, I would say. It's a lot better to most of the drugs in terms of toxicity. So let's say, for example, multiple sclerosis patients are injecting them at least 3 times a week during many years, for all their lives. So, I mean, it's something doable. So maybe that is something you can pursue. Okay, so I think, is there any other question for Monica? If not, okay. So I think we are going to go to the, not the last one, but the one before talk, and is Dr. Tamayo. Changing a bit gears here, we are turning less biological, more physical, and exploring cancer cells through the lens of physics, the role of optomechanical technologies.
Okay. It's a pleasure to be here, I think, in this conference with these very well-recognized experts in cancer. So today, I would like to try to give another perspective about cancer that is from the point of view of physics, that maybe can provide you a novel understanding on the disease and maybe can offer novel therapies and novel diagnosis tools. One of the things that characterize life is motion. Everything that is alive is moving, and this occurs at all levels of complexity. So when I would like to you can pay attention to this human guy that is running. Basically, all the mechanobiological circuits that occurs in life, you can see in this example. So when you are running, basically, you are feeling the ground force on your feet through mechanoreceptors that produce biochemical signals that are processed by the brain, producing another biochemical signals that is going to trigger mechanoactuators. That means it's going to activate the joints, the muscles, in order to make the following stride and not fall over. So that happens also at smaller scales, and in particular to the cell. So basically, the structural integrity of the cell is determined by the cytoskeleton, and in particular, the actin cortex. So in the actin cortex defines how soft or how hard is the cell with respect to external force in the microenvironment. And this is something dynamic. It's continuously remodeling in order to tune the mechanical properties of the cell to the mechanical properties in the microenvironment and also to generate forces. This is a key theme for keeping healthy tissue, for keeping homeostasis. And one of the features of cancer is that disrupt everything or every equilibrium in the tissue. And in particular, there is a loss of equilibrium in the mechanical forces. So cancer is really complex, but I guess that most of you use these cancer hallmarks to try to define common features in different cancers. It becomes to be recognized that also you can define physical hallmarks of cancer that can be associated to the biological cancer hallmarks. So, in particular, in mechanical properties. So, what you are going to find when the cell transforms in a cancerous cell is that it's going to produce a remodeling of the cytoskeleton, and basically the cells are softer than the normal counterparts. However, the cellular matrix is more dense and is cross-linked, and this makes the tissue stiffer. So you have the tissue is stiffer, but the cells are softer. And what I would like to pay attention to today is one of my probably my favorite cancer hallmarks, that is the regulation of the cancer metabolisms, that from the point of view of mechanics, what you are going to see is that the cells have more energy, more power, due to the ATP hydrolysis, that is going to be really important for the regulation of the cell growth and the cell proliferation in cancer, and finally for invasion and metastasis. So it's important to study these cell mechanical properties in cancer. So what we propose here is the use of optomechanical technologies. Basically, just a simple way to study what is optomechanical technology is basically you have always a mechanical sensing element that wants to feel the force in a living system, but to feel the force, you need to know what is the motion of these devices. So always in physics, there was a nice marriage between optics and mechanics in order to develop mechanical sensing elements that are read out by optical cavities or optical interferometric techniques. And this is basically has given nice things in science. I think using a giant interferometer and a mechanical element was detected the first gravitational waves that was predicted by Einstein. And in our case, in a small scale, we use this optomechanical coupling for studying the small fluctuations of nanomechanical devices like silicon nanowires, in which our precision is about 10 to minus 15 meters. That is the size of a proton, that what is quite amazing, the sensitivity of these optomechanical setups. So when you want to study the mechanical properties of a system, you can study this from the point of view of stiffness. That means how resist the system to external force, or you can study the spontaneous fluctuations. In non-living matter, the spontaneous fluctuations are known Brownian fluctuations, because Robert Brown discovered this in the motion of the pollen on water. I think he thought there was a life force, but it's basically a force that arises from the thermal energy and from the thermodynamic equilibrium. And in the case of living cells, we are going to have an additional source of energy, that is the energy of life, is ATP hydrolysis. So we want to study living cells. We want to study the mechanical response to external force, and also we want to study the fluctuations that has valuable information about the metabolic activity, or I will try to convince you of that. So today I'm going to show you briefly 3 technologies. One is more or less well known, that is atomic force microscopy, to study the mechanical resistance of the cell to external force that we apply. The other is not very well known in biology, that is quantitative phase imaging, but we use this technology for label-free and non-invasive measurement of intracellular fluctuations. And finally, by using mechanical devices, we are going to show you that the first proof of the existence of mechanical resonance in living cells. As a proof of concept, and don't kill me for this, we need physics, we need easy models. We use a well-recognized breast cancer model in which we have a normal epithelial breast phenotype, we have a cancerous but non-invasive phenotype, and we have a metastatic cancer phenotype. First of all, just a few words about atomic force microscopy. In atomic force microscopy, in our lab, we use a big ball, this is a colloidal probe, to apply forces, small forces, to the cell, and to see what is the deformation of the cell. Basically, that's produced a bending in the microcantilever that is detected optically. Atomic force microscopy has contributed in the last 10 years to the idea that the cancerous cells absorbed than normal cells in different cell lines and even in patient cells. However, there was a lack of standardization in these measurements, and it was difficult to reproduce the same measurements in different labs and in different conditions and in different techniques. And one of the reasons is because in these experiments, the people was assuming that a living cell, something that is so complex, is elastic. That is impossible that it might be elastic, it's viscoelastic. However, to obtain the viscoelastic parameters of a living cell was, from the point of view of theory, quite challenging, and from the point of view of computational methods, it also was quite challenging. We spent some time to develop these computational methods and algorithms in a way that we can extract 2 viscoelastic parameters. If you look at this equation, we are going to define basically the mechanical properties with two parameters that is not so bad for something so complex. One is A0, that is the elasticity modulus at 1 Hz, and beta, this beta is the power law exponent, and we will call here fluidity because it's 0 for elastic material and it's 1 for a liquid, and will be something in between for viscoelastic materials like cells. Using these two parameters, basically we gain a lot of selectivity for distinguishing, differentiating the breast cell lines. And basically, you are going to find, okay, the cancerous cells are shorter. This is something that we knew, but we find differences between the non-invasive cancer cells and invasive cells. And something that I would like to, for me, is quite interesting, is that the non-invasive cancer cells are the softest of the cell lines. They are really soft, and they are also very fluid-like. That means that these cells don't require a very well-organized structures to resist mechanical forces. And probably what I want to link this is with the cancer metabolism deregulation, that probably the energy that you need to keep a very well-organized actin cortex probably can be deviated in these cells for higher proliferation and faster growth. This is something just a hypothesis, but that's the point of view of the relation in which between energy and mechanical properties is here demonstrated, because when we remove the nutrients and the food and the ATP, basically all the mechanical properties of the cells basically overlap in a single spot, in which basically all these cells are really very soft, and probably they keep the minimal structure to be stable. So that means that something that, from the physical point of view, when you have a system, a structural stiffness is fixed here, requires energy and requires, and this is important because in cancer, cytoskeleton remodeling and the regulation of metabolisms are two key hallmarks that basically you can see here that are well related. So when I was preparing this talk, I was concerned about this question about the energy, and I spent, I know, a stupid way, a couple of days trying to find what is the energy cost of life, and what is in evolutionary biology is called resting energy expenditure, that basically are the kilocalories per day that we need to be alive without moving and without doing anything. And basically, well, that's curious, but what you can see here is that basically as the size of the species decrease, there is a logarithmic, there is a power law in the decrease of the resting power. But basically that is described, that was described by Krieger's law. It breaks down for the small things when R is smaller than 1 millimeter. So basically, humans will require between 50 and 100 watts to be alive. In the case of the human cell, basically it's between 1 and 10 picowatts. Pico means 10 up to minus 12. What we are interested is to see how we can determine the metabolic activity of the cell. Or even more, if we can visualize the metabolic activity in the cell, because I believe that this is important and valuable information in cancer. However, when the things decrease, shrink in size, what happens is that the motion, the intracellular motion, basically is not so easy to distinguish between ATP-driven motion or Brownian motion. And this happens at this scale. So that has motivated in the last 8 years, probably, the development of a tool that probably you don't know. It's an optical technique. It's label-free. They don't need fluorescent dyes. It's quantitative phase imaging. And basically what they use is the... but you can't hear me, no? Okay. And quantitative phase imaging that is based in the holography concept, you are able to retrieve the phase of the light of the wavefront light that goes through the cell. And one of the nice things is that all the non-aqueous components of the cells, the dry mass, the phase of the light is proportional to the dry mass, that is the mass of the non-aqueous components, that mean proteins. So our approach was, okay, if you can visualize the dry mass and the proteins are moving crazy, driven by ATP or by KBT energy, we can try to use this technology to imaging intracellular fluctuations. However, when you want to talk about, when you want to measure stochastic signals, usually the stochastic signal that you wish to detect is hidden by other stochastic source of signals, basically noise in the photodetector, the sound noise, electrical noise, mechanical vibrations. So at the beginning, we couldn't see the intracellular fluctuations. And this is the maths. This is probably the things that are boring for you, but that they are very fun for us. We were spending a lot of time to see how we can decorrelate the noise due to the intracellular fluctuations from other sources of noise. And finally we found a method that has been protected by a patent. And basically, this is what we see with this tool. Basically, on the left, you can see cells. You can see the 3 cell lines, and normal cells, to say in easy words, non-invasive cancer cells and metastatic cells. In the middle, there's a Laplacian filter, just to highlight the internal motion in the cells. And on the right, basically, is the accumulated activity with the time. You can see that in few tens of seconds, you can appreciate the intracellular motion. Normal cells are really quiet. You cannot see so much motion. In cancerous cells, non-invasive, you start to see some structures, round-like structures that are moving and a bit crazy. But in the case of the metastatic cancer cells, they are really nervous, and there are many internal structures that we relate to mitochondria that are trafficking in the cell. So basically, in 100 seconds, you can obtain a picture about the intracellular fluctuations and also about the metabolic activity. Interesting things when we have developed imaging processing methods to develop these fluctuations in single cells. This is an NCF7 breast cancer cell that is not invasive. And basically, what is important in the 3 top graphs is that basically most of the intracellular fluctuations are developed in very specialized structures. There are 7 structures that accounts for the 4% of the total area of the cell, and this are going to produce the 60% of the intracellular fluctuation. So that means that the activity is very concentrated in very specialized regions. To distinguish that if these intracellular fluctuations arise from KBT energy, that means that are from thermal forces or are for ATP driving process by motor activity, we analyze the fluctuations in the frequency domain. And in the frequency domain, we know that basically a logarithmic slope near 1 means Brownian motion, and logarithmic slope near 2 means ATP hydrolysis. And you can see here in the logarithmic slope, that is the last graph in these red regions, we can visualize in a label-free manner where the metabolic activity is concentrated. Basically, using parameters like the power of the fluctuations and the logarithmic slope, basically we can phenotype these cancer cells by measuring the fluctuations. And what is more important, also we can locate spatially where the metabolic activity is created in the cells and how increased with malignancy. And basically, we know that there are some specialized structures that expand in area as the malignancy increase. Using different parameters, we can use deep learning methods, and we can differentiate these cell lines with a specificity of 99%. That basically that's not very challenging in this case, but tells you about how we can use fluctuations to study these cells and also to visualize the metabolic activity. Finally, I would like to talk about our last work. This is a different thing. This is a crazy thing that arises from the 50s, in this Eugene Aukerman, he wonders about if living cells can resonate or can vibrate. And I guess that this was in the Cold War part of this history in which people was concerned about killing things, and in particular pathogenic cells, using ultrasound methods. However, this question has remained without any answer during the last 70 years. And to be honest, if you ask to most of the physicists, they will answer that it was not possible that a cell, a living cell, can resonate because it's very complex, it's a liquid with solid-like structures, and also there's enormous dissipations that is going to quench any resonance. However, we are crazy, and we like to answer these kind of questions. And to answer this question, we use something that we used before, that is the stochastic mechanical resonance entanglement. That basically this, said in easier words, is that basically, if you had two tuning forks and you hit the arrow of what the tuning force, and both tuning forks has similar resonance frequency, the sound waves created by one of the tuning forks is going to activate the resonance of the second tuning force, always with the condition that they might have the similar resonance frequencies. So this is what we use to try to answer the question of Eugene Aukerman. Instead of hitting the tuning fork, we use KBT energy. One of the tuning forks will be a cell that will be coupled to a mechanical transducer. So what is the frequency of the resonance of the cells if we don't have an idea about what are? Basically, in theory, everything is possible, so we could model where are the resonance frequencies of different things. And we found that in the case of eukaryotic cells, this would be, the first resonance frequency would be in the ultrasound regime between tens of kilohertz and hundreds of kilohertz. Even this frequency regime can be also, the bats and the dolphins can also hear in this, at this frequency regime. That is quite curious. So basically, we designed a microcantilever having many vibration modes in order to try to catch the vibration of a cell. So in atomic force microscopy, we functionalized this cantilever with fibronectin, and basically we fish this cell from the culture, and after fishing the cell using fibronectin adhesion, we start to measure the Brownian fluctuations of the microcantilever, not the cell. The Brownian fluctuations of the cantilever, there are two kinds of motion. There are pressural vibrations, and there are torsional vibrations, all hit by the atomic bombardment. So we have 5 vibration modes that can probably be entangled to the vibration mode of the cell in case that this vibration mode of the cell exists. And this is the first proof of that. Basically, on the top is the reproduction of a real-time experiment. The green spectra, frequency spectra, is the real-time frequency spectra. The gray with the white line is before attaching the cell to the microcantilever. So you are going to see 5 vibration modes, if I can move. Basically, what you have to do is in the torsional spectrum of the microcantilever, you see this solder. This solder is increasing with time, with adhesion, as it's increasing in frequency. When we develop these computational methods, we can, when we develop this method, we develop theoretical methods that are quite difficult to explain here, but are quite challenging because you have to combine finite element modeling with viscoelasticity equations, and also with fluid dynamics. But finally, can be identified this solder with a cell resonance, and this is the first proof of concept. In fact, we can obtain those two vibration modes. They see that So basically, now that we have answered this question of Eugene Aukerman 70 years later, we are concerned about if we can measure different mechanical resonance in different cell lines. Still, the method is measuring small fluctuations, so the signal-to-noise ratio is not great. However, we start to see differences, and we know how to improve this. So now, in the next few years, we will try to improve this to distinguish what are the mechanical resonance. The existence of mechanical resonance in living cells, I believe that it's going to open new avenues in the understanding of cell mechanics. Most of the people want to use this for killing cells selectively. That's the naive idea. Okay, if the mechanical resonance are different, you can apply ultrasounds, and then you can kill one cell and not another cells. I'm not going to, it's going to be very easy. But for me, what is the core is that basically there is a field that is starting to grow, still in the infancy, that is mechanobiological therapy. So as I said at the beginning of the talk, cancer is not only a biochemical or a genetic disease, it's a mechanical disease. There is a disruption of the mechanical homeostasis in the tissue. We know that a mechanical stimulus can change the tissue forces, and the tissue forces can be transduced by the cells, and this can produce biochemical routes or biochemical signals that can change the mechanical response and can change the mechanical response of the tissue. So if we can play in this mechanical or mechanobiological circuit, maybe we can have we can obtain new ideas about new therapies. In fact, what was surprising for me that already there is a review without, I don't know, based on 5, 6 words, of the use of mechanical stimulus to stimulate living cells. Just for finishing, this is the last slide. I know that we are starving, but we know that to be starved probably protects us of cancer because we don't provide glucose to the cells. Most of you study cancer complexity, all the biochemical heterogeneity, and basically you know everything about genetic variants during cancer progression and during resistance of the drugs. But I don't know, probably you will say that's stupid. But probably what is difficult for you is to find the functional impact of all this molecular diversity in cancer to determine what are the key biomolecular changes that makes the cell, the cancer more aggressive or that can induce metastasis, or what are the key biomolecular changes that make drug resistance. So the best way probably for me, and you will say, okay, that's stupid, maybe is to look at the physical fitness of the cell. If you measure the physical properties, you can identify the final outcome of this biochemical diversity in how the cell grows, how the cell proliferates, and how the cell is resistant by measuring mechanical properties that can be measured not in 24 hours, can be measured in a few minutes. So I hope that this can have some outcome. Let me thanks to all the people in the lab that basically they are doing an amazing work. Today you will have 2 talks in our lab. That is, Sergio Garcia is going to tell you something about how to detect cancer biomarkers with a sensitivity of a single cell. This is quite amazing. And also Alvaro Cano will deep the introduction to quantitative phase imaging for measuring cell fluctuations. And thank you for your attention.
Any question?
Thanks, Javier. I have a curiosity. How much of these properties correlates with temperature, or what would be your expectation?
Temperature. Okay, basically, when we look at the cells, we look at the culture conditions. They are basically at 37, so we don't play with the temperature.
I was referring more to the intracellular temperature. If you change metabolism, many of these reactions will be exothermic, and maybe intracellularly there are small fluctuations on temperature. And I was wondering if
you can define an effective temperature of the cell based on that. However, this will be not take into account that the temperature in physics is quantified in Kelvin, so it's usually the fluctuations by the equipatrition theory is proportional to the KBT energy. So you have to add 273.2 degrees to the. So by increasing 2 degrees, you're going to from 300 Kelvin to 302 Kelvins. So in this case, it's not significant.
So, Javier, how difficult is to translate these technologies to organoids, meaning 3D cultures where you have cells at different levels? I mean, I'm just asking whether.
I expected this question from you.
No,
but you are also, but you are an author of 2 of these papers.
Yeah, I know.
Okay.
Yes. I remember that. No, but so I mean, do you have option to have, let's say,
yes,
set levels
yes.
of imaging?
We have recently acquired new techniques in the lab that will be for holography in 3D that can be tackle this problem of organoids and to try to have access to this. This is really new, so we don't know still the potential. And organoids will be always a limitation, but anyway, I think this, let's see with these new technologies, we can do something about it because, well, they promise that they can do it. So there are some crazy fundings now that we have used to buy this system and to see what happens.
So have you actually looked at the impact of, let's say, some mutations, particularly, let's say, on the microtubular proteins? What is the impact of a specific mutation, let's say, on those mechanical parameters you're interested in?
Yeah, that's a...
You know, I mean, biologically, maybe I was thinking that something that is maybe not very relevant biologically is going to have very deep impact on some of those properties because it's affecting particularly the cytoskeleton, right?
Yeah, I think, okay, this is quite well studied in the case of cell mechanics. The people more or less have dissected all the components in the cytoskeleton that provides the mechanical stiffness. I think probably microtubules are not the main determinant of the cell mechanics. Basically, it's more the actin cortex what determines the cell deformability, which basically also the cross-linking proteins and also that myosin contractility what determines that. So I cannot say you some number about how contributes each component. In our case, we have applied drugs that target myosin and target also actin fibers. We find that the main determinant is of the cell mechanics is energy and actin fibers.
Maybe I can contribute to the answer. So I think the best example and the best papers with a molecule is JAP1. yeah, so there are a couple of papers, we have discussed this with Japan, which, by the way, is the leader of one of the small cell lung cancer tumors, so I think it's a very interesting question. Yeah.
That's the question, yeah.
Hi, I think it's really, really nice when you compare different cells from the same tissue and you see the differences in stiffness, but how different are cells from different tissues? I guess there might be very different levels of stiffness and the structure of the cells and the all the microenvironment, and how this can affect, for example, metastases. So cells are probably less stiff and they are more metastatic, but how different is the cytoskeleton and the microenvironment in different tissues to be a good place to have a metastasis?
That's a good question. Well, first of all, I had to apologize because when you are developing new technologies, you need a model. So the model, I know that is quite well established and probably is not the most interesting for you. However, we are now apply this technology for different cancer cells. I think the lung cancers, we did some interesting study. And basically, the important thing is about when you are comparing stiffness, you have to compare stiffness with basically the same cell line, basically with mutations in the same cell line. That's what makes this interesting. Comparing pears and apples it doesn't make sense. So that's why I'm collaborating with Marcos Malumbres, because I don't know, I have no idea about what is the relevant cancer problem.
Yeah, for example, if you want to apply these physical parameters to therapy, like for example, you say using different stiffness of the cells as a target, I guess that is very important to know differences between cell lines, no? Between different types of cells in the body, no?
Yeah. Probably, I don't know if I understood well.
So if you want to design a therapy based on differences in mechanical conditions, like in the stiffness of the cells or how, like the resonance of the cells.
Basically, what I would like is to have a system, and basically you have cells from a patient, and basically we can characterize these cells from the patient at one stage, and then you can apply your therapy and to see if it's resistant or it's not resistant. Ideally, it would be a serial screening of the same tumor during progression. That would be ideal, but I know that it's not so easy to obtain living cells from the patient during the progression of disease, or at least.
Okay, thank you very much, Dr. Tamayo. I think we're going to move to the last talk of the morning, and it's Álvaro Ucero. Álvaro is going to tell us about how iron uptake is actually modulating the immunosuppression in squamous lung carcinoma.
Hi, everybody. As Javier said, we are all probably starving, and now we used a lot of glucose to try to fully understand all these complicated concepts, so I will try to be brief. I would really like to tell you what Luis just said, that I know what iron does to suppress immune response in squamous lung carcinoma, but we are still on it, so I don't have the answer. I'm here today to represent the group from the Complutense University, led by Luis, in the context of the iLUNG. And as you already know from the talks this morning, we've been talking a lot on personalized medicine. So I think I'm the only one speaking about subtype of lung cancer that nobody mentioned before. I'm not going to go on the classification of lung cancer because I will be the third one doing so, but I want to tell you that this project, this research is focused on the squamous cell carcinoma subtype of non-small cell lung cancer, which is characterized by clinically by being aggressive and associated to smoking history, and molecularly is characterized by high rate of mutations, high rate of alterations, many of them copy number variations, and there are no actionable targets because most of the alterations are in tumor suppressor genes, that there is a loss of function. So this, as today, is not possible to target therapeutically. In this context, one of the recent advances in the treatment of this disease is the incorporation of immunotherapy as in other cancer subtypes. And there are a lot of clinical studies proving this, so this is the standard of care. In advanced stage, depending on the expression in tumor cells, depending on the expression of PD-L1, the patients get either immunotherapy alone or chemotherapy plus immunotherapy. This is in the advanced stage. In the early stage, they get surgery, and depending on the other factors, they can get perioperative chemotherapy or immunotherapy. Immunotherapy in the neoadjuvant setting, so before surgery, is something that is now giving promising results at this stage. So some years ago, Javier Ramos, a PhD student in Luis Lab, started a comprehensive characterization of squamous cell carcinoma samples from 98 early stage tumors. That these are early stage because these are resected tumors. So we got the samples directly from the surgery, and this is also the reason was that we wanted to do a so comprehensive characterization that we needed a lot of tissue. So we cannot do this kind of experiments today from a biopsy, which is a very small piece of tissue. So together with the clinical annotation of each of the 98 patients, we ran a targeted DNA sequencing to know the molecular alterations, targeted RNA sequencing to know the transcriptomic profile, gene expression, and immunohistochemistry characterization mainly on immune markers because we wanted to find links and profiles between the molecular characteristics of the tumor together with the immune landscape. The main objective was to try to implement a kind of personalized medicine because immunotherapy improved the results, but many patients were not responding. So we wanted to have better biomarkers than PD-L1 to select the patients that might respond to immunotherapy. So we did this. This is a transcriptomic profile that we classify with consensus clustering, an algorithm that gave us, I will try to be brief, gave us a classification of patients according to the expression of these genes, and also a classification of genes according to similar expression. So we got 4 groups of patients and 3 groups of genes where we could characterize the, let's say, the taste of each cluster. So doing overrepresentation analysis, we got to the conclusion that the cluster A of genes was enriched in immune system genes, while the cluster B was more enriched or overrepresented, or it had overrepresentation of genes related to tumor progression. So if we check the expression of the different clusters, we could assume that this first group of patients had low expression of immune-related genes and high expression of tumor progression-related genes. So this group of patients compared to the others was showing a colder profile according to this classification that Julia mentioned as cold with less immune response, hot with more immune response, while this other big group had an opposite profile with high expression of immune-related genes and low expression of tumor progression. So this led us to, let's say, name these 2 big groups. The other groups were like a mix, not that well defined. We call this group like pro-tumoral and this other group like pro-immune. Then, taking this into account, we were putting different layers of information according to this multi-parametric characterization, and we found that actually there are immune populations, there are many more density of these immune populations in the pro-immune groups as expected. But we needed this confirmation. And molecularly, this is just a selection of genes that we consider important for squamous cell carcinoma. We found an accumulation of alterations that usually more genetic alterations is usually associated to a pro-immune group of patients, but in this case, many of these alterations are amplifications, not mutations, so they are not so directly related to immune response. So we saw an accumulation of genes, and with this, we could classify these squamous cell carcinoma patients related to these profiles. Something that called our attention was that the amplification of this gene, TFRC, was strongly related to the cold group, to the immune cold group. This TFRC is the transferrin receptor in the cell, which is the main iron importing system in every cell. So we thought, okay, what has to do the amplification of a transferrin receptor and with the immune cold subgroup of patients? So this is where we started to study, because we had no idea before what TFRC does. So TFRC is a receptor of transferrin. Transferrin is the main carrier of iron in the organism, and it takes the transferrin with the iron, it takes it into the cell, and then the iron is going to be, iron is reactive, it has a lot of metabolic activity, so it's usually stored in the cell in a ferritin complex in an inert way, so it's not harmful for the cell, and there is always a small labile iron pool that is free iron. So this is basically how this system works, and we know that like every of these physiological systems in the organism, it's important to keep the amount of iron in a narrow window, because iron overload, it has a lot of issues, and iron defect, it has a lot of issues. So it's important for the cell, for proliferation, etc. What do we know about this in cancer? Well, actually TFRC is in the chromosome 3, and there is an amplification of chromosome 3 associated to the squamous cancer. So this is not very peculiar. It's kind of expected that in 20–30% of squamous cancers, there is this amplification of this arm that includes TFRC. So TFRC was not the only gene amplifying these patients. This we can also observe in other squamous cancer types, in esophagus, cervix, and head and neck. So it's something that might apply to other cancer types. So from this, we thought that, okay, TFRC amplification is associated to a profile of patients with cold tumors. Can this be a marker of maybe a response to immunotherapy? Can this be a mediator of the disease or even a therapeutic target? Those were our first objectives. So this was the starting point of the project of Maria, who is here today, a PhD student. So, okay, what is TFRC doing in these cancers? Can we use it for a clinical application? So first of all, our objective was to describe, okay, TFRC amplification is there, but does this translate to the expression of messenger RNA? Yes. Patients with amplification have more RNA, and not only that, they also have much more protein, which is expressed in the surface as expected. This is human tumors state for TFRC. And not only that, what happens with ferritin as a surrogate marker of that a cell is taking iron? We see increased ferritin also in the patients that have amplified TFRC. Are these tumors taking more iron? So is this TFRC amplification having a functional role in these tumors? For this, we did an iron staining, is not a very sensitive technique, but we, the pathologist, in collaboration with the group of Fernando López Ríos, pathologists evaluated these stainings both in the tumor and in the stroma, because we know that macrophages take a lot of iron, so we expected positivity in the stroma, but maybe not in the tumor. But we actually saw, we do see positive iron staining in the tumor cells. If we quantify this, what we saw is that the patients with amplification of TFRC had significantly higher iron in the tumor compartment, which also applied to the stroma. In this case, is not a significant difference. Then, since we had a full characterization of the immune populations, we wanted to know if these tumors with the TFRC amplification and uptaking iron, do they have less infiltration? And actually what we saw is that the tumors positive for iron had less CD8, CD4, and CD20 cells. So this is actually true. We had the proof that the tumors, the squamous tumors with amplification of TFRC, they were taking more iron, and this was correlating with less infiltration. So thinking, we are a very translational lab, so thinking, can we apply this to the clinic? We thought, okay, if we could detect this amplification in a non-invasive way, that would be a very good way. So we are setting up the conditions to detect in liquid biopsy this amplification, which being amplification is not easy. So maybe there is something we can do with the group of Dr. Tamayo to detect TFRC in the plasma, but we are also working on detecting the multiple copies of TFRC in the tissue. But something that I can tell you is that in the neoadjuvant setting, so in early stage non-small cell lung cancer, this study run by Mareno Provencio using chemotherapy plus immunotherapy in early stage non-small cell lung cancer, TFRC was one of the genes associated to non-response. So the low TFRC was associated to complete responses in these patients. So this is one data that suggests that maybe TFRC is a good marker to predict the response to immunotherapy, at least at early stage, which is the stage where we found this gene. For the liquid biopsy, we are trying droplet digital PCR to try to find an increased number of reads of the gene in circulating DNA, and using a platform of PDX models, we could find that some squamous tumors with many copies of TFRC, we could detect them in a preliminary result. We could detect them by the system, but we need to prove this in human samples. To study the mechanism, we used a mouse line that was isolated from a carcinogen-driven model of squamous cell carcinoma, and we had several lines in which we just modified the gene to overexpress or knock out TFRC, and then we test that actually the knockout cells were knocked out and they were overexpressing the gene, and these were accumulating ferritin, the overexpressing, meaning that probably they are taking more iron. We studied how these cells grow, and we found that while the knockout cells are growing less, the overexpressing cells are not growing more or faster compared to the control. So TFRC overexpression, which is what mimics what we see in the patients, is not really affecting the viability of these cells. What we do know, using a kit to detect iron, is that these cells overexpressing TFRC in this mouse model are taking more iron. So if we take this to the in vivo setting, because these cells have a pure genetic background, and we can inject it in immune-competent mice to study the growth in vivo in an immunocompetent system, because we know that TFRC can be related to the immune response. So we did a subcutaneous xenograft of these cells in these mice, and we just follow up the volumes. What we see? We saw something that was already described, that a percentage of these tumors regress. But what we found in the TFRC overexpressing cells, we found that only one of the tumors regressed. This is an immunocompetent system, so this could be due to the immune system that the overexpressing cells are less immunogenic. This is something we are actually studying, so I don't have an answer, but we do see a relation between the overexpression of TFRC and maybe an impact in the immune response. We know that in this model we are successful overexpressing the TFRC, so this has been proven. So just to finish, some conclusions is that, okay, this amplification of TFRC in patients with these tumors is associated to immune cold transcriptional profile. They have TFRC protein overexpression and ferritin overexpression. They accumulate more iron, and this is related to a decrease in at least CD8 positive T cells. Regarding the role in the mechanistic role, we know that the overexpression doesn't really influence the cell growth. The knockout actually delays the growth. And also, we can see that the overexpression of TFRC is related to less rejections of this model. But can TFRC be a therapeutic vulnerability? Well, what has been done in the field of cancer so far suggests that we cannot inhibit TFRC because it's everywhere. So it's very toxic. There is an antibody that blocks TFRC that went to the clinic. Clinical trials were stopped by the toxicity, and there is one alternative that is the same antibody with a mask, just to try to, this antibody would get to the tumor metalloproteinases would cut the mask, and then the antibody would block TFRC only in the tumor, and this has shown some promising results actually in squamous cancers, but it's difficult to target because TFRC is everywhere. But something that these tumors are doing is taking more iron, and we know that taking more iron can be toxic. When cells have more iron and they increase the labeled iron pool, they don't directly die because there are systems that compensate this increase with antioxidants. So the cells is stressed, but the cells do not necessarily die. But this condition makes them more sensitive to dying for ferroptosis, which is one form of cell death that depends on the accumulation of iron that leads to lipid peroxidation. So one usual way to induce ferroptosis in cells with more iron and more lipid peroxidation is to inhibit this antioxidant compensation mechanism. So this could be a way to kill selectively the cells with amplification of TFRC, more iron, and maybe more lipid peroxidation. So this is the strategy we are following now. Actually, there is this paper from 2019 where they did a proteogenomic classification of squamous cell lung cancer, and they found a group of patients that had this gain in this chromosome 3 that they call redox. They called redox because they were not inflamed, so this would fit more or less with what we see. But not only that, they call redox because they have antioxidant pathways altered genetically, okay? But they also have proteomically enriched glutathione metabolic process, which is one of these systems that compensate the increased iron to avoid dying for ferroptosis. So this is some data that fits very well with what we see. So maybe we have one new therapeutic vulnerability of these cancers. And with this, I'm finished. I would like to thank all the group in Luis' lab, and also, well, especially Maria and Javi, who are the ones leading all this experimental work, but all the team in general, Esther Conde from Fernando López Ríos' lab, and also the funding of the iLUNG, etc. So many thanks. I'm happy to take questions.
Maybe I miss it. Have you used or are you planning to use iron chelators, which are a good tool in vitro and in vivo?
Yeah, we are planning to do so. Actually, we are starting a collaboration with Richard Posemato in NYU, who is an expert on ferroptosis, and this is one of the approaches that he suggested, because they are being used more and more in the in vivo setting. Actually, there are there are even food for the mice that, yeah, they can have. That's the thing. There is a lot of research on iron, and we can detect iron in many ways, but it's not easy to interfere with the.
Actually, I was thinking right now that, I mean, if we look at, let's say, patients with hemochromatosis, they do have a specific sign on the immunological system. They do have a phenotype.
I don't know, and we don't really understand this because we see the iron both in the tumor compartment and in the stroma. So we are not sure that the availability of iron is really changing for the immune cells, for example. But yeah, if you have this condition, there should be more iron there. So, but I don't know. To be honest, I didn't check.
And I guess you have never seen this signature of iron accumulation in other kind of lung tumors, right? Because iron accumulation is typical, for instance, in colorectal cancer and many others, etc. It correlates with a lot of aging phenotypes, so it's a very bad thing, right? So you have never seen that in lung adenocarcinoma, small cell, neuroendocrine lung, etc.
There are some works on, for example, on ferroptosis as a therapeutic approach, but not that much. We know that different tumors respond different to ferroptosis inducers, but usually these works do not make a direct relation to iron presence in the sample. So, and it's also different having iron in the microenvironment than taking iron. So in a cancer without increased TFRC, probably all the cells are able to take iron in a similar level. So I wouldn't really compare just a tumor with a lot of iron with a tumor with a specific amplification of this receptor.
I guess you have taken out iron levels in the same.
This, I thought, is data that we would need to retrieve from the clinical sheets, but
I'm sure we got that.
Yeah, but I tend to think that if there is this difference, it would be recorded somehow. Yeah. We need to check because.
I'm sure, I mean, we got at least serum ferrum and likely ferritin and transferrin and so on. It would be also interesting, I suppose, to see what happened in other tumors with the amplification on the same spot, which, I mean, let's say what happened on those tumors with whatever amplification there, or with PI3K amplification, which is a gene very nearby. So I suppose that would be interesting, right?
Actually, we are also setting up the fish for TFRC to really see and count the copies in situ in the tissue. And once we get this, the idea would be to check in head and neck and cervix squamous cell carcinoma, because that would be the way to, again, link this amplification to the infiltration of immune cells in other tumor types.
Hi, Alvaro. I was wondering, because most of the redox therapies, they used to be a bit toxic, and perhaps lowering the dose could help to a potential therapy. So it would be great to identify some marker in the membrane of these cell lines, like try to deliver on a specific ADC. This is something that do you have in mind to explore?
Actually, there is one other drug using TFRC as a Trojan horse just to enter the cell, but as I said, there are, all cell types have TFRC, so...
That's the point, like, the receptor is widely expressed, but if you see the synergy in squamous cell lung cancer, could be other receptor used as a Trojan horse, as you said? For example, for a glutathione peroxidase inhibitor or whatever, like...
We can check, we didn't do. Also because, as Luis mentioned, this is the amplification of a whole arm of the chromosome, so there are a variable list of genes that are amplified a lot. So we would need to think to look for something apart from this. And actually this we can do better in other cohorts where we have the whole transcriptome, because in this specific cohort we did a panel of genes for the transcriptome, which is 400 genes, and we don't have everything there. So if what you are suggesting is not in this list, we will not see it. But we can check for a receptor that is co-amplified or co-overexpressed with TFRC. That's a good idea.
Okay, thank you, Alvaro. I think it's time for lunch. I think we are having till 3:30. Lunch is just outside, so thank you.